Hello, everybody. Welcome to this meeting. This meeting is very, very important this century. The topic will be artificial intelligence, CRISPR gene editing, and computational COVID-19 diagnosis. So I will be coordinating the meeting, me and Süleyman Aşır hocam, and he will be speaking after me. Thank you very much.
Hello, Mehmet hocam. Greetings from Cyprus. Welcome, everybody. Thanks for the this kind of nice organization and then starting speech. As you know, the artificial intelligence is influencing the future of almost every sector and every person on the planet. And artificial intelligence has emerged as the driving force behind developing technologies such as big data, robotics, and Internet of Things. And it will continue to do so for the immediate future. So I am very excited to listen to the ideas and studies of the valuable scientists in this organization on this subject, which are CRISPR gene editing and COVID-19 diagnosis, which is very, very important nowadays and in our near future as well. So first of all, I would like to invite our first speaker, İlker Gelişen, Dr. İlker Gelişen, for his speech related with interaction of RNAs with coronavirus as spikes. And before that, let me introduce Dr. İlker Gelişen. He graduated from Hacettepe University Faculty of Medicine in 1977. After specializing in anesthesia and reanimation in Germany between 1978 and 1984, he worked in various positions, including general manager in the pharmaceutical industry in Turkey for 30 years. Since 2013, he has been teaching basic medical sciences, anesthesia, and artificial intelligence as a lecturer at nearest university. His special interests are digital transformation in healthcare and drug discovery and development. And he is the Turkey representative of the International Federation of Associations of Pharmaceutical Physicians and Pharmaceutical Medicine. Okay, this stage is yours, İlker hocam.
Thank you very much. What I actually wanted to present you today is something I will be talking about the most famous disease what we are facing during last almost 2 years, the coronavirus. And I started dealing with artificial intelligence, and whenever you start dealing with artificial intelligence, it's clear you need to start dealing with coronavirus. Everything we do is going to the direction of dealing with artificial intelligence at the moment. Next slide, please.
İlker hocam, there's a problem with this sound, maybe with your plug in the computer.
Can't you hear me?
We can hear you, but it's giving a strange sound.
But what can I switch on the mic?
We cannot hear you, hocam, now.
Can you hear me better? Is it better now?
It's again problematic, hocam. I don't know if you have another headphone.
I will try it. Just a second. Give me a chance. I mean, let me. Now?
Yes, hocam.
Can you hear me better?
It's better, I think, now. Yes. No, hocam, we can. Yeah.
Is actually the artificial intelligence my love artificial intelligence and the diseases. Please, the next slide. Here you see the one before, please. Here you see the two famous universities from Ankara. On the right side, you see the Hacettepe University, and on the left side, you see the Ankara University. Both cover the field of medicine. In 1970, when I started, maybe a physician, and started attending the Hacettepe University, the building you see on the left side is the morphology building of the Ankara University, that the students of the medical faculty come and start training. The building behind is the hospital. It was being built. On the right side, I started everything, and everything was totally different. The time we started learning about the microbiology, our lecturer told us there are viruses, some of which are called as coronaviruses, and he also added, on the safe side, they are not dangerous to us. And I put a note on the lecture notes. I don't exhibition because it's not related to the healthcare. But this slide.
İlker hocam, I am so sorry.
But then the next slide, please.
The sound is not good, so
You cannot hear me.
we can hear you, hocam, but it's just giving sounds.
I actually do not have anything else.
Maybe it's a kind of internet problem, so maybe
I am trying to do my best.
it seems to be an internet problem. Okay, we are so sorry about the internet connection problem of Dr. İlker Gelişen. So we decide to continue with the next speaker, Dr. Abdullahi Umar Ibrahim. And later on, we will try to do some corrections, and then maybe İlker hocam will join us again. So let me introduce first Dr. Abdullahi Umar Ibrahim for his talk titled with Computer-Aided Detection of Diseases. He received higher national diploma in biochemistry from Nigerian Institute of Leather and Science Technology in 2013. He obtained Master of Science degree in bioengineering from Cyprus International University in 2016, and he obtained PhD in biomedical engineering from Nearest University in 2021. Currently, he is a research assistant in biomedical engineering at Nearest University. His research mainly focuses on deep learning application in healthcare and CRISPR technology. Okay, Dr. Abdullahi Umar Ibrahim, the stage is yours. Welcome, Abdullahi. Your microphone is off.
Okay,
Okay.
let me first share my screen before I start the talk.
Yes, please.
Good evening once again. It's a great pleasure for me to introduce this topic, application of artificial intelligence in medicine, which is the main title that I'm going to talk about today.
Abdullahi, please make your presentation full screen, please. Okay.
Okay, I'm going to talk about the application of artificial intelligence in medicine. And as you all know, the field of artificial intelligence has transformed since the invention of the internet. Because of internet, like now we can make a lot of diagnosis very easy, even with the COVID-19 pandemic that has been bothering us in the last 1 to 1.5 years. So irrespective of that, I'm going to discuss about application of artificial intelligence in detection of diseases such as tuberculosis and pneumonia. And in pneumonia, we also have different types of pneumonia, such as conventional pneumonia and COVID-19 pneumonia. In respect to that, so I'm going to discuss, first of all, I would like to define what is artificial intelligence. So artificial intelligence is just the behavior of computers to mimic human beings. So the field of artificial intelligence has some branches such as machine learning and deep learning, which you are going to see. So machine learning is just to program computer to learn without explicitly being it's just to give computer the ability to learn without being explicitly programmed. So in machine learning, we have different categories such as supervised machine learning, unsupervised machine learning, and reinforcement learning. So in supervised machine learning, all we do is just to label data. Assuming we have a car and a cat, we'll label them and tell them this is a car and this is a cat. Then we use models to train the network so that it will be able to distinguish or discriminate between the two levels. So mostly in this research, I'm going to talk about two types of level, such as positive in tuberculosis, as well as general pneumonia. I'm going to talk about positive cases and negative cases. And when it comes to COVID-19, I'm going to talk about more than 2 levels, which you'll see in the coming slide. So in supervised learning, we just label data, and we have different types of machine learning, such as support vector machine, random forest, and decision tree. Unlike supervised machine learning, unsupervised machine learning, in this we don't label the data. We just allow computer to find patterns in the data. So that's the difference, the main difference between supervised and unsupervised. Because supervised, we are supervising it so that it will work the way we want it. But unsupervised is just that the computer is supervising itself so that it will find pattern in the data to be able to categorize them. So most of the unsupervised machine learning are rule mining technique and clustering algorithm. So clustering is just for computer to say, okay, we are going to cluster students. Let's use Near East University. They will say that we are going to cluster students in one cluster and lecturers in one cluster. Or assuming we have different nationals, then the computer will say that, okay, those in Nigeria will classify them, those in Turkey will classify them, those in Cyprus will classify them. That's the main explanation of unsupervised machine learning. While reinforcement learning is just the training computer to perform better at the given task. That's why, like most of reinforcement learning has to do with gaming technology, where computer will learn to play games so that it will perfect itself, just like the game of chess. In the recent years, a machine learning that was trained was able to beat the best chess player in the world. So this is to show you that the computer can learn very well, and it can learn effectively and accurately. So all of these models that I talk about, or types of artificial intelligence, we call them shallow artificial intelligence. They are shallow because they have only maybe one hidden layer. So unlike deep layer, in deep layer we have different types of layers and most especially the hidden layers, because the more layers you have, the better. So just look, for example, assuming if you are going to filter a water. When you are filtering a water, when you use one filter, the filter that you are going to get is not going to be as clean as possible. So what if you have different filters and different size? When you are filtering, you are going to get a lot of the water is going to be cleaner. So that's applied to artificial to deep learning, and the deep learning is inspired by the functionality of our brain, how our brain take neurons and process information. So this is what drive scientists to come up with deep learning. So as you can see here in deep learning, we have an input. You input your images. Let's say we are classifying tuberculosis into positive and negative cases. So after you apply the image as an input, the image will go through these hidden layers. These hidden layers, we call them filters. And after it goes through these hidden layers, it's going to produce an outcome. And in this outcome, we use a probability method to show that if the output that we want, it has to be between 0.5 and 1, the probability of 0.5 and 1, and the other one that is not the outcome is going to be less than 0.51. So compared to traditional machine learning, which has one layer, neural networks that have more layer perform better compared to network that have a single layer. So I'm going to explain this when I'm trying to explain the deep learning models in the next slide. So the most popular machine learning models, deep learning models, are AlexNet, which is developed in 2012, GoogleNet in 2015, ResNet in 2016, and also BGGNet. These are deep learning models that have been tested before, and these models have performed in different competition, and most of them won the competition according to the year. So as you can see, the recent one that won the competition is ResNet in 2016. So having talked about machine learning, so now we are going to talk about transfer learning, which is another core field that I work on. So transfer learning is just to transfer what is trained in one model to another model. So as you can see from the image, it's just like this bird trying to transfer something to her offsprings. So this is the case of transfer learning. As you can see here, we train a model in model A. So the algorithm that we use to train this model would repurpose it and use it in model B. Why do we do this? Is because to increase the performance, because a model that has been trained is already has already established a connection and is more accurate and efficient than a model that is going to start from the beginning. So that shows that you can't compare someone that is starting afresh with someone that has experience. So that's the logical way to explain transfer learning. That's why you see in this image, if you develop a model from scratch, it's going to have less performance compared to using transfer learning model. So in this presentation, I'm going to talk about mostly about AlexNet because it is the model that we use in most of our research. And AlexNet has 8 layers. We have input layers, we have 5 hidden layers, and we have 3 fully connected layers. So these layers, they just process images so that we'll get a desired output. So the main objectives of all of our study is just to train AlexNet model so that it will be able to classify between cases or images, let's say positive and negative, or COVID-19, non-COVID-19, and also to compare with performance with other neural networks such as ResNet, SqueezeNet, MobileNet, and also to compare the performance with human, because what is the aim of training computer if it's not going to outperform human beings? Because human beings make a lot of errors, so in order to see if our model is better, we have to do this comparison study. So artificial intelligence has evolved in medicine, and it has shown a lot of potential and promises. And in the last few years, it has been used for detection of lung cancer from CT scans, as also as cardiac health from electrograms, and classifying of skin lesions from images, and also the identification of retinopathy from the eye. So this to show you that apart from image classification, artificial intelligence is also used in medical record and prediction of disease. As you can see, in the last year, there has been a lot of studies that predict or forecast COVID-19 pandemic. So this is just to show you an example or a list of the application of artificial intelligence in medicine. So having said that, I'm going to discuss about the core advantage of our study is just computer-aided detection. So we use computer with models so that it will help in detection of disease. So in healthcare, most of the medical images are used in this research in order to classify them into different classes. So in the case of early detection of disease, especially when it comes to cancer, detection of cancer at an early stage is to improve patient survival rate. So you can't compare someone that was just diagnosed early and someone that is diagnosed late. So that's why artificial intelligence is is aided in detection of disease at an early age. So the core of this study is going to be pneumonia, such as general pneumonia and COVID-19, and also different types of pneumonia, such as bacterial and fungal pneumonia, and also tuberculosis. So tuberculosis is a disease caused by a bacteria known as Mycobacterium tuberculosis. As you can see from the image, this is how it looks. Mostly it can only be viewed using the microscope. And we have two types of tuberculosis. One is latent, which is dormant, and active, which cause symptoms and can be transmitted. But this dormant, it can be transformed into active under conditions of normal temperature and pressure. And this, as you can see from this image, the bacteria is slender in nature. It is rod-shaped, and it is strict aerobe because it can't survive without oxygen. So in the absence of oxygen, the bacteria is going to die. And another important quality or characteristics of this tuberculosis, it is acid fast, which is mycolic acid. This acid fast make it like if you dye it, you are going to see the bacteria easily. So tuberculosis can be transmitted just by sneezing. That's why people that are active, they used to be isolated so that they will not transfer the disease to other people. And the symptoms of tuberculosis include weight loss, coughing, and fever, and also night sweat. So in order to detect tuberculosis, we have some conventional techniques that we use. One of them is tuberculin test. The other one is a Nelson Spotting smear test. This is a test that we normally use in general hospital. We just dye the slide. We'll add some colors and dye it so that we are going to see if the bacteria is present or not. Another most important technique, which is also common, is the chest X-ray. And another one is interferon gamma release assay. And recently, scientists have developed a CRISPR-based biosensor, which use the gene of the bacteria and also target in order to detect the presence of the tuberculosis. So this is a topic that we publish in Journal of Infectious Disease, is automated detection of Mycobacterium tuberculosis using transfer learning. So the reason why we decided to train this model is because this tuberculosis is very small and like it has a low contrast background and irregular appearances, as I show you in the image. So scientists, especially microbiologists, find it difficult to differentiate this bacteria, except if they use a high technological, sophisticated microscope. And another issue is to count this bacteria is very difficult because of the overlapping of the bacteria. And sometimes in a hospital that have a lot of cases, this is going to be very, very tedious for the microbiologists to detect this bacteria in, let's say, more than 1,000 number of cases. So what we do, we label our image into positive and negative, and we pass the images into a model. This is how the models look like. This is the computation of the model. As I explained before, we use AlexNet, and it has 8 layers, the input layer, then the 5 hidden layers, and the 3 fully connected layers. After we pass these images into this model, is going to tell us whether it is positive or negative. So after we train the model, as you can see from this image, this is the slide image of tuberculosis, and this is we use this slide image and train it in a computer, and the computer was able to discriminate between healthy and infected cases, and we obtain accuracy of 98.73, sensitivity of 98.59, and specificity of 98.84. So this is the journal that we publish the work, the Journal of Infections in Developing Countries. So this is all about tuberculosis. So the next study that we publish is pneumonia classification using deep learning from chest X-ray images during COVID-19 pandemic. So before I get into the main details, I would like to explain what is tuberculosis and how many people are infected. So before COVID-19 pandemic, we have more than 150 people that are infected with tuberculosis annually, and majority of them are children. And this tuberculosis can be, this pneumonia can be caused by different types of pathogens such as bacteria, fungi, and also viruses. So in terms of the data set that we use in this study, we use chest X-ray images. As you can see, the one from the left, as you can see, because of this white spot that you are seeing, that shows that the person is positive. And here, because there is no white spot, that shows that the person is negative. So we obtained these images from a study conducted by Kamani et al. In 2018. The images are made available online, so they are free. Anyone can get the images and use them to do different types of study. So we label the images into positive and negative cases, and we pass them through AlexNet model. And at the end of the day, we have a classification. So this is how we label them into negative and positive, and we pass them through the model, and the model was able to discriminate between healthy and infected. And this is we have a training accuracy of 96.43, 94.05 as testing accuracy, 98.19% as sensitivity, and 95.78% as specificity. This shows that the model was very good and accurate in detection of detection of pneumonia. So this is published book in Springer in the early 2021. So the next topic that I'm going to talk about is the one that has to do with viral pneumonia. So I know that we are all conversant with the COVID-19, but let me just give a brief summary about COVID-19. It's caused by SARS-CoV-2, which is severe acute respiratory coronavirus, and is one of the family of coronavirus family that have caused global health in the past. We have MERS, which has caused epidemic in the Middle East, and we also have SARS-CoV-1, which have caused an epidemic in 2002, and it's also initiated from China. So the recent COVID-19 started in Wuhan in China, and from that is separate, but the World Health Organization only declared it as pandemic on 11 March 2020. 2020. So this is a very distinct type of viruses. It has a spike glycoprotein and it is only made of RNA. Unlike other organisms that are made of DNA, these viruses are made of RNA. So coronavirus can be transmitted from infected animals to human, and human also can also transfer the COVID-19 via sneezing and coughing, and it can also be transmitted when someone gets in contact with infected places or handles such as doors handles, windows, toilet bowls, and light, or maybe if someone sneezed on top of a table and someone came and put his hand there, he can get infected. So these are the symptoms of COVID-19 pneumonia, which includes fever, fatigue, nausea, diarrhea, and aches. So in order to train artificial intelligence model or deep learning model, we need a data set. So we obtain part of the data set from GitHub. We obtain 153 images. Then we also obtain COVID-19 normal X-ray images and non-COVID-19 viral pneumonia from Kaggle, which is also online repository. It's also available, anyone can access it. And we also obtain different images from Kaggle as well, and the one that we use in the study conducted by Kamani et al. So these are the type of images that we have. We have normal cases. We have normal cases. We have COVID-19, non-COVID-19 viral pneumonia, and also bacterial pneumonia. So there is no way human beings can be able to discriminate between these three types of pneumonia because they look almost the same. So no one can say this is bacterial pneumonia, this is viral pneumonia, or this is COVID-19 pneumonia. So that's why this work is a very the first of its kind that try to classify these types of pneumonia. So what we did is we split the data set into 70 for training and 30% for testing. So after we conducted image augmentation and processing, which we tried to increase the number of data set by cropping, by zooming, by changing the color. And after we trained the images using deep learning models, and the image we were able to discriminate between positive and negative cases. So this is how the model window look like. All we do is we input the algorithm. After we import the algorithm, then it passed and it trained the model, and after training we conducted testing, and from that we can test different images, even from images that are not within our domain. So this is the most important aspect of artificial intelligence. So first of all, we try to see if we can discriminate between non-COVID-19 viral pneumonia and healthy, and we obtain high accuracy and high sensitivity. Then we try to see if we can also distinguish between bacterial pneumonia and healthy, and also between COVID-19 and healthy. After that, we said, okay, why not differentiate between COVID-19 and not COVID-19 viral pneumonia? Because all of them, they are viral pneumonia, so it's going to be very tricky and difficult for human beings to discriminate, because human beings can easily discriminate between bacterial pneumonia and healthy, non-COVID-19 and healthy, COVID-19 and healthy. But to discriminate between these two, because all of them, they are positive, so it's going to be difficult. So after we conducted this study, we obtained high performance. Then we said, okay, why if we try to see if the model can be able to discriminate between three? So now we increase it to three classes, and we also have high accuracy. So, but the more category we add, the less accuracy we obtain. Then we said that, okay, this last study is the first study that is conducted in the literature that has to do with COVID-19, non-COVID-19 viral pneumonia, bacterial pneumonia, and healthy cases. In this aspect, we have 4 classes. Initially, scientists only reported 3 classes, so we are the first to report these 4 classes. So, we were able to publish this work in expert system, and So in details, the detection of tuberculosis and pneumonia was made possible using AlexNet architecture. And in this study, we use pre-trained AlexNet model, which means the use of transfer learning. And this transfer learning has shows that it can aid scientists in detection of mycobacterial tuberculosis, pneumonia, and COVID-19. So, and also this model has shown that it can relieve this the hospital can use this model in order to confirm the presence of tuberculosis bacteria or pneumonia or COVID-19 pneumonia, and also relieve the workload of radiologists when it comes to pneumonia and COVID-19 cases. So these are my references, and thank you very much for listening.
O zaman soru olarak.
Okay, thank you, Dr. Abdullahi Umar Ibrahim, for your nice talk and nice presentation. Any questions? I guess, let me check questions, but I haven't seen any questions. But okay, if anybody needs to ask you, they will reach you easily from your mail. So we would like to thank you again for your contribution to the field and the contribution to this seminar. Thank you very much again.
Thank you very much. It's nice presenting.
Okay, now I would like to invite our next speaker, Dr. Ahsen Özcan, for her talk related with principles of CRISPR and gene editing. Very interesting topic. So before that, let me introduce Dr. Ahsen Özcan. Dr. Ahsen received her undergraduate education in biological sciences from Middle East Technical University. After a short period of experience in the United States with Fulbright, she completed her master's degree at Sabancı University. She completed his postdoctoral studies at MIT in the field of CRISPR-Cas. Following his doctoral studies at the Max Planck Institute in Germany with a TÜBİTAK International Doctoral Scholarship. After completing her postdoctoral studies at MIT, she returned to her homeland and conducted research at TÜBİTAK Marmara Research Center, and is currently working at Helmholtz Institute for RNA-based infection research in Würzburg, Germany.
Thank you for the nice introduction.
Hocam, welcome.
Thank you.
Okay, the stage is yours.
Thank you. And first of all, thank you for the invitation for this nice symposium. And I will be mainly responsible for giving the basics of the CRISPR-Cas biology and recent advances which leads to the CRISPR-based applications. Next slide, please. So this is really basic, the central dogma, which is the essentiality of the life. And what we can see right now is CRISPR-Cas based technology is now goes into the DNA level and mRNA level. And still, like, there is nothing going on with the protein engineering yet. Next slide. Next slide. And so the main question is, like, where does CRISPR-Cas system comes? And it is actually the natural defense system between the viruses and prokaryotes. And as you can see, viruses are the most crowded biological entities on the earth. The poor bacteria has to defend itself toward the viruses, just like us, and which leads to a very divergent immune systems by the prokaryotes, and CRISPR-Cas is one of them. Next slide. And I really want to specify not only the CRISPR-Cas systems, but also the other defense systems, because each of these defense systems actually have very big potential, which can go to biotechnology or diagnosis or applications. And I think the biggest place is the discovering of these defense systems and then adapting them to the adaptations. And as you can see, the virus initially needs to attach itself to bacteria. And bacteria developed like several strategies by making like receptor modifications, which can inhibit the attachment of virus, or phage infection injection system blocking pathways like Z system. Or alternatively, when the phage is completely infected, infect its genetic material, it could be inactivated by restriction modifications, which is actually, as you can remember, from the restriction enzymes. And CRISPR-Cas systems, newly found systems like disarm, breaks, or Argonaut. And if the infection is in an unbearable stage, a cell can lead itself to program that by toxin, antitoxin, or abortive infection systems. Next slide. And I will be mainly talking about the CRISPR-Cas adaptive immunity, because this CRISPR-Cas is a bit different than the other ones, because it can store the information of the infections. It's an adaptive immune system. And when the first infection comes, adaptation modules, Cas1, Cas2 enzymes, you can see in the like small little squares. It can cut the pieces of the viral DNA and then puts inside of the CRISPR array. And next slide. And when the second infection comes, this CRISPR array becomes an immune. It becomes an immune library, and with the combination of the Cas enzymes, they create interference. Next step. And you can see the like CRISPR loci, which is composed of repeats and spacers, and then next to it there will be Cas genes. Next slide. And if we would like to describe what's the function of this genome is, CRISPR loci works as a virus library. You can find the pieces of the viral DNA. And then Cas genes are mainly nucleases, and you can think that it's scissors, genome scissors. Next slide. And there's like multiple stages that I briefly mentioned, but each step is quite important for the immunity. And each step is actually leading to a kind of new applications in every day. So the first step is adaptation. Previous slide, please. Yeah. The first step is adaptation, which the viral DNA is cut and then put into the virus library. And this part, actually, the adaptation part, is actually now used for the infection recordings as an application. Next slide. After adaptation, we call it expression, which is like transcription of the whole CRISPR array. And by the help of the Cas nucleases, these very big pre-CRISPR RNA is processed into small CRISPR RNAs. For the Cas9 case, we say it like guide RNA now with the tracer. And combination with the other Cas nucleases, which is expressed next to the CRISPR loci, they create interference, and which leads to the degradation of the virus. And next slide. And I think interference is one of the most interesting part, where the nucleases are actually coupled with the CRISPR RNAs, and then which means that they have a now GPS system by the CRISPR RNA, and nuclease is getting guided through those specific RNAs. And I think this is very remarkable because you now have the guided nucleases. Next slide. And as I mentioned at the beginning of my talk, the virus and the race between bacteria creates enormous systems, and CRISPR-Cas is one of them. And CRISPR-Cas is also by itself has enormous diversity. And this is the end product of evolution. So it is also a very big job to classify those systems because every new year, new systems are coming, and based on the demand, we try to characterize more nucleases. But so far, the characterization is class 1 and class 2. Class 1 is very much based on multiple effector complexes, which is like CRISPR RNA is coupled with more than one nucleases or proteins. It's like multiple proteins come together and create the complex. And for the class 2, which is a single effector protein, which means that the adaptation, expression, and interference is done by just one protein. And they are called single effectors. And they're one of the most popular guys in the field because they can do everything by themselves and they have more applications as compared to class 1. Next step. And this is very representational of how actually they look like. Class 1, as you can see, multiple proteins, bunch of proteins come together around the CRISPR RNA skeleton. And for the class 2, just as you can see from Cas9 or CPF1, just one protein couples with the RNA. Next slide. And as I mentioned, CRISPR-Cas is highly divergent system. At 2015, there was 2 classes, 5 types, and 16 subtypes. And when we come to earlier today, next slide. Next slide. It is 2 classes, 6 type, and 33 subtypes. And every year, this is getting updated. We see new members in the family. But I think what is quite remarkable in this tree is this is like a family of the CRISPR-Cas enzymes and systems. Next slide. We have quite interesting things going on in the class 2. Because type 2, where the Cas9 comes from, type 5 or type 6 has, they have single proteins, but different features as well. And this year, there was also another groundbreaking discovery was found. And even in class 1 systems, we can also see the single effector proteins. Next slide. And I want to briefly mention about the Madonna of the field, which is the Cas9. Next slide. And the most popular one is the Cas9 from Streptococcus pyogenes. This is the one which is discovered almost first and also adapted for the genome editing. And its protocols are highly developed and very much easy to do. And this Cas9 had been created the most problem, but also the Nobel Prize itself. As you can see from Streptopyogenes Cas9, there is RuC and HNH domains. Those are the 2 nuclease domains which are responsible for cutting the double-stranded DNA. Next slide. And as many people can see, Cas9, mostly used by the Streptococcus pyogenes, Cas9 is used, but the other Cas9s are also available. But every Cas9 has, since every protein or nuclease has different pump sequences, the pump sequence which is responsible for distinguishing self from non-self. And it is quite important for the genome editing because the guide RNA that you're designing is highly dependent on the pump sequence. And Streptococcus pyogenes uses just NGG. 2G is enough to find, but whereas others have like a multiple different pump sequences, which is reported so far. Next slide. And I also would like to very briefly mention about the CRISPR Toolbox, because one part is discovering the basic science of the CRISPR, which leads to new tools actually. And in the community, the other part is very much interested in developing tools out of the CRISPRs. And what we can see so far is CRISPR is almost everywhere. And it started with the gene editing, and with the deactivated Cas9, which is like a making mutation in the active side of the ROC and HNH, you can program Cas9 for just binding. And with this dCas9, the application called CRISPR-I, which leads to gene regulations, opening, closing, or alternatively, you can like add more proteins to the Cas9, and which can lead to trans effector activities or epigenetic modifications. And then by this programmable protein, like a guide RNA, which programs the Cas9, you can do chromatin imaging or single base changing by putting the by making the Cas9 nickase, which is just making one mutation in the Cas9, and addition of like putting base editors together with the Cas9. Base editors are also approved recently, and then they're highly used in the medical field. And by the characterization of the more CRISPR systems, we come up with the RNA targeting enzymes like Cas13. New, very unpublished paper also suggests Cas7, 11, and type 3 systems, and which leads to developments into the RNA therapeutics field and also diagnostics. And it's also very much very popular to work with the chromatin topology and chromatin imaging together with the DNA targeting systems like Cas9 or CPF1 or Cas12 systems. Next slide. And I would like to also emphasize a bit more into CRISPR diagnostics, which become highly popular during the pandemic, is it was actually discovered with the Sherlock, the CRISPR-based diagnostics, which means they detect the viruses at very low level from the patient samples. And these detections were based on mainly collateral cleavage activities of Cas12 or Cas13. But what we can see right now also, like development of the CRISPR chips or type 3 systems also adapted for the disease detections or nucleic acid detection platforms. Or I can happily also say that the most recent detection CRISPR diagnostics also suggests multiplex way of detection of the COVID variants with the Leopard system. This is highly new advances. This month it was published. And one very nice thing is these systems, CRISPR adapted systems, have been adapted for even very low level detections with the by using the lateral flows or fluorescence techniques, which can lead to very home-based or very point of care, or which can be very useful for the places which doesn't have access to the hospitals as well. And I would like to end my presentation, and I'm waiting for the questions. Thank you very much.
Okay. Thank you very much, Dr. Ahsen Özcan, for the very interesting presentation about CRISPR and Cas9. Thanks a lot. So when we check the questions, I haven't seen any question in the comments, but I may ask a question for you. I mean, that's an interesting, very interesting subject for me as well. I want to ask about the CRISPR babies. What do you think about the babies? I think Nana and Lulu, twins in China, they are engineered as HIV resist gene. So, but is there any problem using with this system to engineer the human genome like this, or what do you think? Maybe in the future, is it possible to maybe COVID-19 resist babies is possible like this? Or I want to learn your ideas about that.
To be honest, I'm completely rejecting this Nana and Nulu. I think it's quite unethical to use these systems in human societies. And I mean, the science sometimes develops so fast, and people cannot really catch, or the regulators of this where the science cannot catch. And there is a stop point for using it. Like, you can develop products out of it, diagnostics and the other things. But I think it's not very ethical to make humans or armies out of CRISPRs. This is really nonsense. And I think you can also think which kind of world you would like to live in if you would be able to edit your own genome. And this is highly dangerous and highly unethical. I think my position would be completely I would not approve.
Of course, I agree with you completely. Okay, thanks a lot again for your contribution and for the very nice talk. I would like to meet again in another meeting.
Thank you very much for the invitation.
Bye. Okay, we are going to continue with the next talk. Will be given by Zubaida Said Ameen with the title of Predicting of Single Guide RNA Activity for CRISPR-Cas12 System Using Deep Learning Models. So before that, let me introduce Zubaida to you. She received a Bachelor of Science degree in biochemistry from Bayero University, Kano, in 2010. She obtained Master of Science degree in bioengineering from Cyprus International University in 2016. Currently, she is an academic staff in biochemistry department, Yusuf Maitama Sule University, Kano, and a PhD student in biomedical engineering at Nearest University. Her research mainly focuses on deep learning and CRISPR technology. Okay, so Zubaida, welcome, Zubaida. Your microphone is off.
Thank you for the introduction.
Yes, Zubaida, I think it's okay now. Welcome.
Okay, thank you. So I will be presenting on the topic prediction of sgRNA activity for CRISPR-Cas12a, that is the CPF1 system, using deep learning models. Based on my outline, I will give an introduction on the CRISPR-Cas12 system and machine learning and deep learning. Then I will explain the prediction of the CPF1 activity using deep learning models. And finally, I will conclude. So for the introduction, the CRISPR-Cas system is now the most powerful tool that is used for gene editing. This is because of its simplicity and efficiency. It utilizes just two components, a guide RNA and a Cas enzyme, to cut any target DNA. The guide RNA directs the Cas enzyme to the target DNA. After binding to the target DNA, which has matching sequence, the Cas enzyme will generate double-strand breaks. these double-strand breaks can be repaired by the cell using either of the two mechanisms, and this can result to gene knockout or gene knock-in. So the issue here is the activity of the guide RNA, because it has a possibility of binding to other non-intended sequence in the genome that can disrupt normal gene function. That is why it is very important to predict the guide RNA activity. So in this presentation, I will explain how we can utilize deep learning to predict the activity of the guide RNA. Briefly, I will explain the most commonly used CRISPR-Cas systems, the CRISPR-Cas9 and the CRISPR-Cas12a, which is the CPF1. We can see in this figure, the CRISPR-Cas12a, for the guide RNA, it has only CRISPR RNA shown in green, and for CRISPR-Cas9, it has a CRISPR RNA as well as the tracer RNA, so the guide RNA is more complex. And in terms of the PAM requirements, the CRISPR-Cas12a requires a T-rich region shown in pink, and as for Cas9, it requires the NGG PAM. So, and the mechanism of cutting, DNA cutting, it's staggered cuts in the CRISPR-Cas12a, and it will generate sticky ends, while the Cas9, it generates blunt ends. So based on these features, scientists can select which CRISPR-Cas system will be more suitable for his research. So the reason why we need deep learning in CRISPR-Cas system is because of these two issues. We have off-target effects and on-target effects. For the off-target effects, we know that the guide RNA can bind to a matching sequence on the genome, but also because the genome has billions of nucleotides, the guide RNA can bind to also other similar sequence on the genome, and that will lead to unintended cutting of DNA. So it is important to predict the activity of the guide RNA. And also another issue is the on-target effect. We know that the guide RNA will bind to the matching sequence on the target genome, but we are not sure if it is going to generate double-strand breaks. So deep learning can be applied to predict the activity of this guide RNA. Machine learning, it involves the use of a model to transform input data into output. So a machine learning model can learn using data and algorithm. So the model will understand patterns of the data and use that knowledge to make predictions on future examples. So deep learning is that subfield of machine learning, but deep learning uses neural networks. A neural network is made up of layers that transform inputs into output. And we can see that in the figure. Each layer in the neural network has neurons that are interconnected to each other. So this arrangement of layers and neurons process input data into output. And the convolutional neural network is a type of deep neural network, and it is used for processing images. And it is made up of two parts, as we can see in this figure. It has the feature extraction part as well as the classification or prediction part. So the feature extraction part consists of convolutional layers, while the classification part is made up of the fully connected layers or dense layers. So the input to a convolutional neural network is an image. An image is presented as a matrix, as shown in this figure. So convolution operation is basically matrix operations. The input is multiplied by another matrix, which is called a filter, to extract important features from the input. And these extracted features can be passed into the pooling layer, and the most significant information will be extracted from the pooling layer, and these features will be passed to the fully connected layers, and the fully connected layers, we utilize this information to make predictions. So we are going to use convolutional neural network in this presentation. As I'm going to explain earlier, we used a convolutional neural network to predict the guide RNA activity because it is possible to represent guide RNA in the form of this matrix. So after genome editing with the CRISPR-Cas system, there will be double-strand breaks, and these double-strand breaks can be repaired using non-homologous end joining, but the process is not 100% perfect. It can lead to some insertion or deletion of nucleotides. These are called indels. So these indels can be detected after the experiments using some techniques like the sequencing techniques or next generation sequencing techniques, or indel detection using by amplifier assay. But all these techniques, as we can see, they take hours and days, and they require high instrumentation, and they are very expensive, and they require expertise also. So our aim is to predict this indel frequency using deep learning models. So with our model, we can be able to predict the indel frequency within seconds. And this indel frequency will tell us if there was double-strand breaks, and it means our guide RNA is active. So if the indel frequency is high, it means the guide RNA is active, and if it is if it is low, it means the guide RNA is less active. So our main is to utilize these deep learning models to predict indel frequency, since it corresponds to guide RNA activity. And then we develop different types of models and compare their performances. Also, we design guide RNAs that can detect SARS-CoV-2 gene. For the methodology, the work is basically put into 3 sections. The first is the data processing, where we obtain our data set and then convert the data into a format using one-hot encoding. And for the models, we utilize the 3 different types of models. We use the multilinear regressor, which is a machine learning model, and a convolutional neural network, and a hybrid, that is a combination of convolutional neural network and support vector regressor. Then we compare the performances of the model using Spearman correlation in order to determine if the model is doing a good job or not. So the format of our data, it consists of guide RNA sequences and their indel frequencies, and the data was obtained from Kim et al. The data consists of 1,251 guide RNAs that are arranged in this format, and each guide RNA has its corresponding indel frequency. So we trained our model using this data so that it can be able to predict indel frequency for any given guide RNA. So first, we need to convert our guide RNA into a matrix. As I've explained, the input to a convolutional neural network is the matrix. So the guide RNA were converted using one-hot encoding. So each of the 4 nucleotides in the guide RNA were presented as one-hot vectors. And finally, we obtain each guide RNA as a matrix. Then we utilize this matrix as the input into our convolutional neural network. And then first the input goes into the convolutional neural layers. The convolutional layers will extract features that can be used for the prediction. And then these features will be passed into the fully collected layers, the dense layers. Then the dense layers will use these features to make predictions. And the last layer will produce our output that corresponds to the indel frequency. And in order to build a good model with good predictive function, we need to optimize some hyperparameters. So in our work, we optimize the number of convolutional layers and the number of dense layers. And then we optimize the number of filters used for each convolutional layer. And also we optimize the number of neurons in each dense layers in order to achieve a model with good predictive performance. And we use the Spearman correlation to compare the performances of our model. The Spearman correlation basically shows relationship between the model's predicted output and the actual output. If the predicted output by the model is close to the actual output, the Spearman correlation score will be high. So if a model has high Spearman correlation, it means it has good predictive performance. And based on the Spearman correlation, for the different models, we can see that the hybrid models achieved best performance with 0.748 Spearman correlation. Also, we compared the performance of our model with state-of-the-art models, that is Tyndall and CPF1, and our model outperformed these models. Then we used our trained model to understand the importance of a nucleotide at a particular position. So if a nucleotide is important at a particular position, it will appear darker in red. As we can see in A, at position 1, C is the most significant nucleotide, so it is very important in the first position, while T is less important in the first position. This means that C is the favored nucleotide at position 1 in the guide RNA sequence, and T is disfavored nucleotide in the position 1. We compared this result with a web tool shown in B, which is KP logo, and we obtained similar results. It also shows that the C, cytosine, is the most important nucleotide in the first position, shown in top, and the bottom, thymine, is disfavored nucleotide. This is because it has been observed that thymine disrupts interaction between the guide RNA and the CPF1 enzyme. So this shows that our model is well trained and has the ability to understand the importance of each nucleotide in the guide RNA sequence. And also, we utilized our model to predict the activity of guide RNAs that can be used for detection of SARS-CoV-2 gene. So we designed guide RNA sequences that will detect SARS-CoV-2 gene and used our trained model to predict their indel frequency. So based on the indel frequency score, we can select and choose the best guide RNA that can detect COVID-19. So this shows that our model also has application in detection of COVID-19, because this will save time as well as experimental costs. And our work was published in this journal, Alexandria Engineering Journal, for the hybrid model. And also we developed different CNN models for prediction of guide RNA activity. In conclusion, I would like to say that it is highly important to design successful guide RNAs for gene editing, because CRISPR-Cas system is now the most important tool used in gene editing. Scientists can now edit genes of humans and plants, and also study genetic diseases and study other gene functions. But the success of the system depends on the activity of the guide RNA. So it is very important to design good guide RNAs. And therefore, deep learning can help to overcome the challenge of guide RNA design and also maximize the activity of guide RNA. Thank you. I would like to take questions if
I'm so sorry for the sound. Yes, I think I forget it. Okay, muted. So thank you very much, Zubaida, for your good presentation and then for your supports. So as I check the comments, no questions, special questions. So we thank you again for your contribution and for your presentation. Thank you very much.
Thank you.
Thank you. Okay, now we continue with the next speaker, Professor Mehmet Özsüz from nearest university, with the talk titled CRISPR and CRISPR-Cas-based biosensor design, another interesting topic. Before that, let me introduce Professor Özsüz to you. Mehmet Özsüz is a professor of biomedical engineering, nearest university in northern Cyprus, and he has graduated from the chemical engineering department, Middle East Technical University. He got his PhD degree in analytical chemistry from Faculty of Pharmacy, Ege University. He has been a post-graduation work in the USA and has been a visiting researcher at the Lancaster University, UK. Dr. Özsöz's research is based on developing electrochemical nucleic acid biosensors, and he has published over 135 research articles in peer-reviewed international journals, 1 book editor, and 4 book chapters with H factor 45 according to Web of Science. His researches have been focused on drug DNA interaction, foodborne pathogen detection, and mutation-related disease detection. COVID-19 detection based on CRISPR. He is also a recipient of Turkish Science and Research Establishment TÜBİTAK Science Award in 2008 on extraordinary work done internationally on the electrochemical, enzyme, and DNA-based biosensors. His recent work focuses on CRISPR-Cas gene editing, artificial intelligence, and detection of CRISPR-Cas9 mediated mutations. We would like to welcome Professor Özsüz to the stage. Welcome, Hocam.
Okay, thank you very much, Süleyman Hocam. So could you see my presentation?
Okay, this stage is yours, Hocam.
Okay, thank you. Okay, once again, I thank you to organizing committee and the Near East University that, you know, this gives us this opportunity to give this meeting. Now, my talk is about CRISPR and CRISPR-Cas-based biosensor. Well, I put my the first slide as the CRISPR picture. As you could see, this one, the one that we have done one paper, and this is the schematic way of expressing the CRISPR. As you could see that later I will talk about what is the going on that one. So this is the double-strand DNA, that this is this gene, you know, and it is Cas9 enzyme. And then we use the gene that we firstly double-strand break make, and then after that by using the single guide RNA, and then after this step, you know, what we are going to do now, there was 2 methods. That one of them that non-homology and joining. The other one is some gene is donor to the system. So we have used this method. So let's go to the talk, you know. So outline of the talk, as you know, the clustered regularly interspaced palindromic repeats as CRISPRs, and mechanism of CRISPR-Cas community in bacteria and archaea. But earlier speaker also mentioned about it, but I will just mention a little bit about that. While in CRISPR-Cas systems in genome editing, detection of coronavirus by CRISPR method, and detection of CRISPR-Cas mediated mutation using electrochemical biosensors, results and conclusion. So I would like to give you some information about, you know, my first major area was the analytical chemistry, electrochemistry, then come to the CRISPR and microRNA, and then finally is the artificial intelligence. So it means that we combine together, but the molecular biology is not my main subject. So please forgive me if I do some mistakes about this one. So as you know that the first thing come out in the literature in the CRISPR was the Japanese scientist from Ishinowa, and then from that one came the Mojica, is the Spanish scientist, and then the CRISPR named by this Mojica and Yanand, And then the most important the development in the Barango is a food scientist. The first experiment has been done. Then to 2012, the paper done by the Doudna and Carpenter, and this paper got the Nobel Prize for the chemistry 2020. So these are some short way of talking about. What are the CRISPR actually? The CRISPR is a defense mechanism of bacteria. So how did it happening? Just I will explain it. This is the locus organization. Well, at the first site, genes coding proteins, and these CRISPR arrays. What are the CRISPR arrays? There are palindromic repeats, and then just before this one, there are the spacer, And what is the, how the things going on, When the first time the invader, the virus come to the bacteria, so they injects the double-strand DNA, and the first time the Cas1 and Cas2 complex, cut it, the part of it, and then as you could see here, this is double-strand DNA, and then this red, the double-strand DNA is put it in the CRISPR locus, as you could see this one. Well, this is on for the what I say. This is the red virus part, and the other green virus, as we say, and just between is the spacer, So, what are the other mechanisms that going further, Then these are the expression and maturation, and as you could see that this is the DNA first, then it's converted to transcribed to the RNA, then on this RNA that it is cut down by the enzyme Cas nuclease, by cut, and then what we get is CRISPR RNA. And as you can see, this is the red one is or green one is coming from the virus part. This is the spacer RNA, coming from the virus part. And then when they combine with the enzyme, then what we get it, CRISPR RNA, Cas RNP complex. Okay, then the interference step, what happens in this, the same virus comes the next time, then what happens? So this is the virus again comes in, and then this is the CRISPR RNA, and then when they match it, then this nuclease protein is activated and cut it, the double strand break. So this means that target degradation is happening. So this was the case, Okay, now again, this is the same showing up by Doudna's paper. So this is it's usually the genome of the bacteria, and then as this is the cleavage again, and as you can see, that is the CRISPR RNA, and then when the same virus comes next time, or as you can see, this is the green virus comes next time, and then this one is degraded by this way when they match the guide RNA with the system, and then this is the way that how the CRISPR system in bacteria archaea is functioning. Okay, now this was in the bacteria side. The other thing, this is for double-strand DNA, for instance. You want to precisely cut it down. Then what you gonna do that? You need to have the guiding RNA. See, this is the guiding RNA has two RNA. One of them is the CRISPR RNA, and the other one is the tracer RNA. And then the Doudna and the Carpentier, that synthesize this one is outside in the laboratory, and they use it in the gene editing system. What are this one? When the matching with the guide RNA, then this activated, and then what happens? That double strand breaks is happening, But what we can see is the PAM, PAM is just, it's very, very important. Protospacer adjacent motif, what I usually, is very important, difficult to understand for the students, that what I really explain them, PAM is something that, for instance, you want to go from the city of Izmir to Istanbul. Just before coming to Istanbul, you see the sign of what? 500 meters to Istanbul, then comes the 200 meters to Istanbul. It's just like the, same analogy can be applied that to find the the real part, I mean, the matching site, you know, that we have to have, you know, the PAM, you know. So what happens, you know, after the cutting down, this DNA in the cell is not free to any, that they want to combine together, you know. Just what happens in that case when combining by, you know, randomly non-homologous and joining, what we say it, deleting some part of it and addition of some part, you know. So indel formation is forming, you know, but this one is prone to the mistakes, you know. So the next one is this one, homology is directed repeat, you know. So what we do, you know, we add for instance, the gene that to the system, you know, and then we convert this one to the intended gene, you know. So this was the story about this one. And then what was before gene editing? There was also their methods, you know, many methods, for instance, meganonucleases, zinc finger nucleases, and talon, and the lastly, the CRISPR technology. So it means that both of them, when the double-strand DNA is break down, you know, as you could see that non-homology and joining, and then the other one is the homology direct repeat, you know, as I say, you know, that you add additionally this template to the system, you know. So we did the same thing, you know, in our experiment in biosensor design. So as you could see that Ameen Carpentier and Jennifer got the Nobel Prize in Chemistry 2020, you know, by the paper that the 2012, you know, the paper. And then, so now we discuss about, okay, there are three types, actually, that is most important and we use in application, you know, Cas9, Cas12, and Cas13, you know. What are the difference between them? Okay, as you know that this is the double-strand DNA, and this is the guide RNA. This is made of CRISPR RNA and tracer RNA. And what are in this case, the PAM, you know, is NGG, you know. So what kind of double-strand breaks? And this is blunt ends. And then we discussed the first has been found, and then application has been done it. The second one, this is the CPF1 or Cas12a, you know. Everything is same, but in this case, the guide RNA is just only the CRISPR RNA. There is no tracer RNA. There's only one RNA is needed in this case. And different from the Cas9, this is the PAM sequence is TN. So what happens, you know, when the breaks down, it is the target is still, you know, double-strand DNA, and then you get staggered, you know, cuts in this one, and then this one and this the CRISPR-13, you know, Cas13 will be used for the application of detection of the COVID-19 or other virus detections. So this is the most important part, you know, CRISPR Cas13, as you could see, this is the enzyme, and as you, what is the target in this case? Is the target is the RNA, you know, this is the CRISPR RNA. So when they matching the system, you know, this red one, the spacer part, to the target RNA, you know, then this triggers the enzyme to cut it down, you know. So when this cut it down, you know, it can cut any RNA around this one. And again, after this one, you know, after this matching the system, you know, again, the enzyme cut it down any single-strand DNA around it, you know. So it means that in Cas12 is single-strand DNA, and the Cas13 is the RNAs. So this will be using in our, the applications, you know. So, for instance, the one review that we have got, you know, recently related to Cas13. So, as you could see, that's the CRISPR Cas13, you know, this is RNA in solution, and then when they match it, you know, the target RNA, you know, as you could see that before targeting, there is nothing, you know, we could see this is all RNA is in the right position. So when the CRISPR RNA matches the target RNA, then this triggers this Cas13 enzyme, and then it begins the cutting down. So it will be used for our purposes, you know. So it means that there is this already been done, talk about before speakers, early speakers, Ahsen hocams. So there's one which is Gothenburg and done the Sherlock. The other one is the detector, the Outnight all. As you could see that in both cases, in this case, the double-strand DNA, and then by the recombinase polymerase amplification, then the double-strand DNA. And again, with RNA, with RT-RP, as you could see that, that we can obtain the complementary DNAs. And then both of them is DNA, then transcribed to the RNA. So this is the Cas13, and this is the guide RNA. So when the match is when it's activated, by this complementarity, it cuts any RNA around it, As you could see, there's a black one. This is a reporter molecule, binded together with this RNA. This is, as you could see, this is a black one. This one is what we call quencher, and there's this one report to fluorescence, So when these two parts are close to each other, there will be no sign of anything else. When they begin to cut it down, this is activation of this one, you will see that since these two parts are separated, detection becomes, This is the mechanism most important that is detection of the viruses or DNA. In detector system, DNA, again, then it goes with the recombinase polymerase amplification, the DNA. And again, Cas12, again, in this case, this reporter molecule is made of the single-strand DNA. And as you can see that after the binding and matching the guide RNA to the target, then it will cut any single-strand DNA around it. And this molecule, reporter molecule, when it's cut it down as the usual, as the Sherlock, that you will be seeing that this fluorescence one. This is the sign that is important, Okay. Mm-hmm. So, detector, as you can see, detection of the COVID-19, nasopharyngeal swab and then extraction of RNA, what happens in that SARS-CoV-2 E gene and SARS-CoV-2 N gene and RNase P humans, in this case, after isothermal amplification, So this is your target, and then this is the Cas12 complex, okay. Then when this matches this one with the target, then it cuts down any single-strand DNA around it. And then when this cut it down, so this sign of the fluorescence, you could see it. And then when you put it in the lateral flow system, that you can also see by the visual way. And this is the lateral way. When this is the single-strand DNA, this is the FAM reporter, FAM part and bulletin part, and then it is cut it down due to this activity of the Cas12. And when this is negative, you see this sign only, and it is positive, you can see this sign. This is the way that short way of detection of COVID-19. So now this is also another important thing has come out later in the literature that I want to mention about it. Inhaled CRISPR treatment targets influenza and SARS-CoV-2 infections, This is by the Blanchard et al. And Nature Biotechnology. We have seen that CRISPR-based treatment to stop the replication of the flu virus and the virus that causes COVID-19 in mice, So what happens in this case? Messenger RNA technologies, code to form the Cas13, which will destroy the part of the RNA genetic code. So guide RNA in this case tells the Cas13 protein where to attach the virus RNA and destroy. This is really interesting development that I want to put it down here. And then the other new things, but already, it's like CRISPR, now it's come to the anti-CRISPR, What CRISPR is, is a defense mechanism of, what can we say, the bacteria. And can we say about that the anti-CRISPR is the defense mechanism of the virus, So how these things happening, This is again in a paper, in Nature Microbiology in 2018, you know. Well, for instance, in CRISPR, you know, as you could see here, you know, this CRISPR-Cas enzyme, you know, degrade the double-strand DNA of the phages, you know. Whereas, you know, in the anti-CRISPR system, you know, the CRISPR system is blocked, you know, and it's no more active, you know. So it means that the virus are alive, you know, as you can see that. And in this case, again, with the CRISPR-Cas systems and this anti-CRISPR gene system. So this is really is important for the, at least, you know, for the food sciences, you know, for you want to keep the foods, you know, in shelf, you know, for 1 year or sometimes 2 years, you know, how can you eliminate the bacterial growth that, so this can be controlled by this anti-CRISPR mechanism. So not only this in food science, this can be also for the off-target problems in CRISPR technology, you know, that's, you know, can eliminate and can detect the importance of it. So what does the anti-CRISPR proteins can do it, you know, can block CRISPR activity, you know, in many ways. For instance, inhibiting CRISPR RNA loading in this ways, and then inhibiting DNA binding, and or inhibition DNA cleavage. So this is the something that's related to anti-CRISPR technology and it's growing interest in the literature. Now let's come to our talk about the biosensor genome detection. You know, this work then has begun about in the United Kingdom in Lancaster about 2016, but we did a lot of things, but due to so patent issues that this came about this year, take about 4 or 5 years, you know, that's just the beginning. It was, you know, 2016, you know. So we have a group of people from the Lancaster University, people from Ege University, and people from the Institute of Technology, Izmir. So it was three kind of university that we were working. So now, what is the things that we are doing now? As we explained before, that this is our gene, you know, the murine CIZ1 gene, you know. So we want to get a mutation to the system, to the gene, you know. So what we did, you know, we first of all we designed our the spacer, a guide RNA, you know, and then when we introduce to the cell, you know, what happens, you know, that it will cut it down, you know. When after this double strand breaks, you know, as you could, then there were 2 methods. One of them is the non-homologous directed repeat, and the other one homologous directed repeat. So we use the second method, you know. So this is our target sequence. Okay, so we want to get, you know, what the mutation to the system, you know. What is the condition in this case? The homologous directed repeats. The parts should be homologous. This one and this one, and again this part also homologous. So it's similar to the systems. The only difference is the point mutations. Okay, now, so after a double strand break, so what we did, we had a single strand oligonucleotide, about 100 nucleotide, you know, that contains, as you could see, that, you know, point mutations. So when this happened, and then the final is the product then made PCR. After the PCR, we just sequence the PCR product, and then we are trying to get, you know, whether this mutation has happened or not, you know. This is our aim, is this one, you know. How can we make it, you know? So so we obtain this one, this is the final mutation, but we can also detect by the method, by the sequencing, but this one, okay, is the usual way, but how can we detect this one? By biosensor technology. So it means that, so we use the PCR product of this one, and then accordingly, the electrochemistry we applied, you know, the electrochemical system. So how did we do this one, you know? For instance, this is the product that we get, you know, after mutation got it, you know. So what we do is in this electrochemistry part, you know, in this PCR product, we select, you know, the target, and the probe. This is special type for this electrochemical part, you know. So it means that this is not only 3 or 4, this is normally 20 base pair. As you could see that this is the probe and the target part. This is complementary. These are synthetics. One we'll get later on. So how these things going on electrochemistry, you know? Well, this is any DNA in electrochemistry has 2 signals that is apparent, you know. There is one guanine signal at 1 volt. The other one is the adenine signal is around 1 to 1.2 volt. What are these one? As you can see, this is analogy, this potential to spectrophotometric method is the lambda, you know, okay, wavelength. The other one is the current is response to absorbance. So it means that we know that at 1 volt potential, the guanine gives, you know, the signal. And as you could see that in the single strand, you know, the guanine are free to reactions, to oxidations, compared to the double strand hybrid form. You know, in the hybrid form, the guanine is bonded by 3 hydrogen bonds to cytosine. So it means that, as is the chemistry show that the guanine is more reactions, and then it means the signal is higher than compared to the double strand here. So these are the main parameter that we play in this technology. Now, what are the other tools that we use in our system, you know? And we know that the guanine has a 3 hydrogen bond with the cytosine, but and again the cousin of it, you know, we can say inosine, you know, has 2 hydrogen bond again with the cytosine. The difference between two components is that the one is the oxidizable. It means that give reactions, but the one that is inosine, yes, you know, it doesn't give any reactions. So it means that when you're designing your probe, for instance, instead of using the guanine, so we will be using inosine. In that case, we'll get no response, you know. It means that when we get the no response at the probe, it means that when they combine it again, we'll see that we'll always get guanine in the complementary one, we'll get a guanine signal. So this is the basic principle. So as you can see that this is single-strand DNA system, that this is inosine, you know, and that due to this one, you know, there is no response. This is 0 response. When you see that after hybridization, you know, due to this guanine, we'll have response. It means that it is more easy to differentiate between the single strand and double strand, you know. Okay, this is our big. So again, that how do we design our the spacer? Protospace dynamics this way, you know, we get it. This is the protocol that is obtained from the company, and this is the 100 base oligonucleotide we put it, and then these are the 2 mismatch point mutations that we introduced to the system, you know. Okay, and this is the PCR product, and again, then when we get the sequence of the PCR product, you see this is the mutation one. So our aim is to prove that this is the real one. This is which is already been mutated, you know, by the biosensor technology. Okay. So what we did, you know, we did the mutated target sequences. Then we have the probes. As you can see that, you know, the I, you know, is used inosine, you know, instead of the guanine, you know. So it means that when we do it, you know, when we get the signal for this one, no signal, but after the hybridization, you know, so due to this C's, you know, we'll get the guanine. So again, this is for the mutated part, and we get also the synthetic, the wild type, you know. Again, this is the target, and this is the probe, you know. And the probe is always contains, you know, this inosine. This will give the 0 signal, you know. So this was how the design of the biosensor is being done, you know. So this is the procedure that we do it, you know. We use a nanomaterial carbon nanotubes, you know. Then we put interaction with the probe, And then after immobilization of this one to the electrode surface, then we hybridize with the target solution. So as you could see that, there is no signal for the probe, So it means that this is for instance, this is the full match. What do you mean by full match, Probe and the target, which is full complementary to each other, you get the highest response. Whereas, if there is a probe and the mismatch, then the response is not high as the full match, and the non-complementary is the lowest one. So this was the parameter that we involved this technology. And then this is how we can get, for instance, this is the signal of the probe, since the ionism has no response, Okay, this one, you see the ionism, there is no quanine. When this hybridized means that full match, when it's full match, this is the signal, full match. And this is the probe plus the mismatch is only this height, and the probe plus the non-complementary, you get this response. So you can differentiate, probe, full match, mismatch, and this is our basic displacement. Now, so what are the other parameters that we work about this one? Let's go to the optimization of the probe and target concentration. So what we do actually, this is when the full match is the high response, and then this should be the highest compared to the other ones. Respect to that, these are the smaller. So difference between these two and this one has to be high. So this was the 5 ppm is the optimized one. And what kind of other parameters we work? Hybridization buffer, and this is again the optimum one, and the washing buffer and washing time. These are the optimized things for the experimental part. So what are this one? For instance, we did first of all, we have to do it with the synthetic one, synthetic one. Okay, synthetic target, synthetic probe. And then after this one, optimize this one, we use this one, big one is, you see, red one is the PCR product, which is denaturated. It becomes a single strand, So it means that, so the strand in both cases are the same, This is probe, as you could see, due to the ionism, this is 0. And you could see the synthetic one, you see this one, and this, and still with the mismatch smaller, and the smaller again with this one. And when we use the PCR amplicons, denaturated PCR amplicon with the probe hybridization, you get this highest response. So it means the trend in both cases the same. So we decided that we can prove that this is the mutation has been completed. So conclusion, well, what we can do with the CRISPR Cas, precision of genome engineering, and also what we use in this system, Cas9 and homogenic repeats with electrochemical sensors. And then this method is alternative to DNA sequencing and RFLP methods with reduced cost and assay time. So this method can also be further developed for the detection of other types of CRISPR-based manipulations, including small insertion and deletion and multiple base substitution generated by the HDR, base editing, or prime editing. So there, I have put also only 4 of the papers that we did recently. The one that is synthetic biology that was that about 1 week ago is published in the American Chemical Society. CRISPR-Cas13 system is a promising and versatile tool for cancer diagnosis, therapy, and research. And this paper also, the Ahsen Hocam is one of the co-author here. And then again, the other, again, clinical biochemistry, we have used CRISPR-based tools, alternative methods for diagnosis of COVID-19, So this is the, again, a group at the Hacettepe University. And this is the other part with the machine learning. The one that you have listened today with the Zubaida, she is my PhD student, And also this is the Abdullahi Umar, he's also my PhD student, but finished 1 month ago. So we did the convolutional neural network for diagnosis of viral pneumonia and COVID-19 alkaline disease. So these were the related papers that we have already published. So Amach, I will be thank you very much for the listeners. And I would like to thank you for universities, the Yakında University, Nearest University, Ege University, you know, I am originally from that university, and then Lancaster University and the Izmir Institute of Technology. And this, the two people from Nicola Copeland from Lancaster University and the John Hardy, Lancaster University, these are two professors already there. And the Pınar Kara, she's in Ege University. She's a professor now. She's my student also. And then also there is Institute of Technology that they created us nanomaterial, you know, carbon nanotubes. That's very, very important. And then Fahrettin Palace in Hacettepe University. Okay, and then I should talk about Ahsen Hoca. Also, she was co-author of the last paper. Okay, thank you very much. If you have any questions, I'll be happy. And if any question that you can ask me later on, that I can answer. Thank you very much.
Thank you very much, Professor Özsöz, for the very nice talk and very nice presentation. So from the audience, I guess there's no question, but if I ask a question, is that okay?
Yes, please go ahead.
Yes. Okay. Okay, that's really interesting to replace guanine with ionosine in the explained prop. I mean, a prop and target interaction. I'm just wonder to learn if there is any non-specific hybridization, how it is affected when we change guanine with ionosine. I mean, is that any
yes
thing related with that one? I mean, any measurement? Have you? Is it?
So we can prove this way. You know, for instance, we do you know, probe signal, you know, which contains, you know, the guanine. So you see the guanine signal, say at 1 volt. You see, it's sure. But if you replace the guanine with the inosine, you know, you see no signals. So this is the proof of experimental proof. You know, that's what we get it. You know, so we can do this way in the experimental electrochemistry. You know. So we begin about this 20 years happened that this technology, you know, being in the Ege University begin it, you know. First I got this from the United States, you know, when I was a postdoc, you see. And then when I came to, then we begin about 1996, the genome biosensor, DNA biosensor. So this is the first thing. And this technology, by the way, we use pencil graphite electrode. This is pencil graphite electrode, very cheap, and it can be used very, very cheaply, you know. So this technology, electrochemistry, is very, very important, you know. Nowadays, the graphene-based, you know, technology that the group of San Francisco, I think that the Aran group, you know, she has already, you know, used this electrochemical technology, and it's promising in the new future. Okay. Thank you.
Okay, thank you very much, Hocam. Thanks again. Okay, now we continue with the next speaker, Associate Professor Sertan Serte from Nearest University, with the talk titled Computational COVID-19 Diagnosis, another interesting topic. Let me first introduce Associate Professor Sertan Serte. Sertan Serte received his Bachelor of Science degree with honors in Electrical and Electronic Engineering from Eastern Mediterranean University. He also did his Master of Science program in Multimedia Signal Processing and Communications at the University of Surrey. Then he completed his PhD in Electronic Engineering program at Queen Mary, the University of London. While he was in the Multimedia and Vision Research Group, he worked in artificial intelligence, machine learning, image, video analysis of facial gestures, and multimodal human-computer interaction. He is currently Associate Professor Doctor in Electrical and Electronic Engineering Department in Nearest University. His main research interests are in the area of artificial intelligence, and machine learning. In particular, he's working on the application of artificial intelligence for healthcare. Okay, Associate Professor Sertan Serte, I would like to welcome him to the stage. Welcome, Hocam.
Hello, thanks for introduction.
Hello.
Can I start now?
Yes, you can start.
Okay, thank you very much. Let me start show. Okay, today I would like to talk about computational COVID-19 diagnosis. Many of you know that the COVID-19 is a recent infection in worldwide, and it has been appeared in 2019. And so in my presentation today, I would like to talk about convolutional neural networks. And then I will give some types of the convolutional neural networks, and then after I introduce convolutional neural networks, I will talk about computational COVID-19 diagnosis. So convolutional neural networks are important because we need these algorithms and methods in order to teach computers or machines to understand or diagnose the COVID-19 on images. So I would like to just give you a short summary and description of the convolutional neural networks. And there are several types of convolutional neural networks, and some of the examples I can summarize here. One of the well-known architectures is AlexNet, and then later they introduced VGG16, VGG19. Then after VGG, Google LENet has been introduced. Then we have ResNet models. We have different ResNet models: ResNet 18, 34, 50, 101, and 152. Then later we have MobileNet version 2, SqueezeNet, ShuffleNet, and DenseNet. These are the different architectures of convolutional neural networks, and these networks are based on convolution process, filtering process, and so they are based on convolution, but they are architectures, and the processing of the images or the learning the image content is different in each of the modes. How we can use these convolutional neural networks in real application, I can give you an example. If you have an image, if you have a dog in the image, just you can input this image into convolutional neural network, and you can get output of different categories. Of course, the output of the convolutional neural networks are the probability values of the classes, and the highest probability gives you the content of the image. So in this example, you have the highest probability of dog class. So we say that since the highest probability is provided for the dog class, so you say that this image contains dog class or dog image. If you look at the architecture of the AlexNet in detail, we can see that we have the 5 convolutional layers, and the 3 fully connected layers. In the architecture of AlexNet, the convolutional layers are responsible for representing the image content, and these convolutional layers are responsible for modeling the image content, or in other words, the convolutional layers are responsible for representing the features of the image. Then we have 3 fully connected layers, and these 3 fully connected layers are used for classifying or calculating the or modeling the classes and separating each class and then producing the output class scores. As I have explained you before, the class scores are the probability values, and after fully connected layers, we are getting the class scores, and by looking at these class scores, we just decide on the image content or the image class. So this is the basic description of convolutional neural networks, and this is the basic example of the usage or the application of convolutional neural networks into the real problems or images. So if you look at in general picture, you can separate the AlexNet into, or the convolutional neural network into, the two main groups. And in the first group we have the image representation block, and image representation block includes the convolutional layers, and in the last block includes the fully connected layers, and this is responsible for classification. Okay, we can go into more detail of the architecture of the convolutional neural network, and we can look at in detail, and then we can see that actually the first convolutional layer includes 11 by 11 filters, 11 by 11 filter sizes, and you have the stride parameters and padding parameters. And if you look at the convolution tool, or on the right side, is more clear. In the second convolution, layer, you have the 5 by 5 filters. In third convolutional layer, fourth convolutional layer, and fifth convolutional layer, you have 3 by 3 filters. Then you just apply these filters into the images or the convolutional layers in order to create the convolutional layers. And then after you represent these, after fifth convolutional layer, you obtain the fully connected vector 1, 2, and 3. Then finally you get the class scores. This is the main detailed architecture and parameters of the AlexNet architecture. Okay. VGGNet is different than AlexNet. VGGNet contains 16 layers. In the AlexNet, it contains 8 layers. So basically, VGGNet is a bigger version of the AlexNet. The architecture of the VGG is bigger than AlexNet, and you have more layers, and all the filter sizes are 3 by 3. If I go into the next slide, you can also use VGG19, and VGG19 is bigger than VGG16 and AlexNet architectures. VGG-Net 19 includes more convolutional layers, and the filter sizes are 3 by 3. So these are the main differences between AlexNet, VGG16, VGG19. We have also ResNet models. In ResNet models, we have 18, and these ResNet types, different ResNet models might include 152 layers. So in the ResNet model, you can have more layers. You can have bigger architecture. So the ResNet model is much bigger than AlexNet, VGG19, VGG16. So these are the main differences. So you can think of that the ResNet might contain more layers than AlexNet or VGG16. So these are the summary of the convolutional neural networks, and mainly convolutional neural networks are used for COVID diagnosis. automatic COVID diagnosis on medical images. So after I have introduced you the basic models, I can talk about COVID-19 diagnosis. It is well known that we have several diagnostic approaches for COVID-19 infection. The well-known test is PCR test, and this PCR test is one of the methods to detect or diagnose COVID-19. We have also medical imaging techniques for diagnosis, and the X-ray images is one of the medical imaging technique, and another technique is CT scans. So there mainly we have two groups of diagnostic systems. So in order to understand COVID-19 infection, so COVID-19 is occurring in our lungs or the patient lungs and develops in patient lungs. So the lung infections can be listed as maybe 13 or 14 different infection types. And the COVID-19 is one of the lung infections among them. So once we have listed all the lung infections, so the AI approach for diagnosis is to just classify different types of infections. If we classify these infections, we can understand that as you can see, pneumonia can be classified as viral pneumonia, bacterial pneumonia, fungus pneumonia, and under the viral pneumonia, we have coronavirus, and then coronavirus can also be divided into COVID-19, SARS, and MERS. So from this picture you can clearly see the scenario here. So we need to determine to classify the viral pneumonia. Then if we detect viral pneumonia, we can have coronavirus. Then we can reach COVID-19. So the X-ray image is one of the medical imaging technique for diagnosing COVID-19. As you know, these X-ray images are mainly used in hospitals, and every hospital includes X-ray images, and then these medical imaging is fast, and the patients can go under X-ray scan, and basically it is a 2D image, which on this image you can see COVID deformation or COVID signs. So the doctors take the X-ray image of the patient, and the doctor or the radiologist just look at for the COVID-19 signs. If they see any signs, they say that okay, this patient is infected by COVID-19. This is how it works. Then X-ray images also shows different kinds of signs on the images, different signs of the infections. So the radiologist should get these scans and check for the signs of COVID-19, or if the patient is not COVID-19, the radiologist or the doctor should decide or should diagnose another type of the infection. So we have different infections. So when you do COVID-19 infection, maybe if you look at for COVID-19, maybe the patient is infected with tuberculosis or maybe bacterial or viral pneumonia. So you should also know these different types of infection. So the main aim is to distinguish or separate COVID-19 from other disease of other infectious actually. So after X-ray images, we have CT scans. CT scans are the 3D images. CT scans are 3D images, and it includes up to 300 2D images of the patient. So it's a 3D scan, and it is better than X-ray images because it shows you a lot of information, more information than X-ray images. It scans the CT scan scans the patient lungs, 3D scans from upper body to lower body. So you can have many 2D images. There is also here the better picture, so you can better understand the CT scan. So we have the 3D body here, 3D human body or the patient body, and then the CT scan scans the lungs from upper body to lower body, and for after scan you can see the 2D images from upper body to lower body. You can take these and extract them, and then you can obtain axial CT slices. In this case, we have the slices or the images, and we have T number of slices, and then according to different brands, different machines, you can get different number of CT slices, and then these slices shows the lungs from upper body to lower body. Then in this way, you can see whole structure of the lung, and you can detect, you can see where is the infection is in the human lungs or human body. So it gives you more details than X-ray images, and it is so in some cases in hospitals, when the doctors or radiologists needs to understand better about the level of the COVID-19 infection, they have to do the CT scan in order to get more detailed information from the patient's body. So CT scans are important in hospitals and mainly used during the pandemic situations. And CT scans are very important to understand fully about the patient's situation. Whether the patient is in a critical condition or whether the patient is in the beginning stage. So this medical imaging is the only way to understand the situation of the patient in detail. So after CT scan, after doing this process, scanning and then extracting the slices, the convolutional neural networks can look at each of the slices, CT slices, and the convolutional neural network processes each of the slices separately, and then tries to give you idea of whether the slice, CT slice, contains a COVID-19 infection or not. So we have two classes here. We have COVID-19 or normal, or you can also increase the output classes. You can say COVID-19 infection, normal infection, and other infections, so that you can pass or you can input each of the CT slice into the convolutional neural network, and the convolutional neural network provides you the output probabilities of each class. And if the output probability is highest for the COVID-19, the slice containing the COVID-19 infection is expected. So this is how they apply machine learning techniques or the convolutional neural network into medical images for COVID-19 detection. In the beginning of my presentation, I have explained you the dog image. I gave example, and I said that if this is if there is a dog in the image, you get the dog, or the highest, the probability of the dog class is highest. So you can change this image and you can put one of these slices, CT slices, and in this case, convolutional neural network will tell you the probability of the COVID-19 infection and the normal class. So by looking at the probabilities, you can decide whether the image contains COVID-19 or other infections, or this slice belongs to healthy person. In conclusion, I would like here, I try to explain you I talk about the COVID-19 detection or COVID-19 diagnosis. So we have a PCR test, we have medical imaging techniques, and this is the in this presentation, the CT scans are the best medical technique, which provides you the more details about the COVID infection or situation of the COVID infection. So the CT scans are the best way of understanding the COVID, and after getting the COVID scan, if you get all slices and pass into the convolutional neural network, you can detect COVID-19 for each slices, and by this way you can understand the location of the infection in human body, in human lungs, and you can also understand the the condition of the patient, whether it is in a critical situation or in an early stage. Because if the person is in critical stage, the treatment is different. If it is in an early stage, is different. So these are, I try to explain you the differences and the main techniques for COVID-19 detection. And thank you very much. Okay, this is my presentation. If you have any questions, you can answer.
Thank you very much, Associate Professor Sertan Serte, for the valuable information and the nice work that you present here. I guess there are no questions now from the audience, so we thank you again for your support. And then
thank you very much. Thank you very much for this opportunity. Thank you. Bye-bye.
Okay, see you. Bye. Okay, now we're gonna continue with the next speaker from USA, Professor Hikmet Budak, with the talk entitled "Application of Multiplex CRISPR in Plants and Use of Machine Learning." So before that, let me introduce Professor Hikmet Budak. He received his PhD degree from the University of Nebraska-Lincoln, USA in 2002. He worked as a coordinator and led buffalo grass genetics and breeding program at the same university, then returned to Turkey in 2004 and served as professor at Sabancı University until 2015. In 2016, he returned to Montana State University, USA, as an endowed chair. He is currently working as a Chief Science Officer at Montana Bioagriculture, Inc., USA. He also serves as adjunct professor at the University of Nebraska-Lincoln, USA. He has over 180 publications and with over 23,000 citations. He is currently working on genome editing and machine learning in plant systems. Okay, so we would like to welcome Professor Hikmet Budak to the stage.
Great,
Welcome,
hello.
Hocam.
Yeah.
Hello. I hope
First
you
of all-
are good. I mean, you are from USA, and the time difference, as you know, that's a little bit difficult for you. But thanks for your contributions and then all the things.
I thank you. First of all, I'd like to really pass along my thanks to organizer, you guys, and as well as the university as well, the Near East University. It's a great pleasure to be here for, first of all, really.
It's a pleasure for us, Hocam.
And I just stayed up all night, but there were lots of good, excellent presentations. So I really enjoyed listening to everybody as well. So today, I listened to everybody. I'm going to talk about that genome editing and so that the machine learning as a breeding tools. But you can see that the picture, I will mainly focusing on that the wheat, which is one of the stable crops in the world. And any examples or any just the research that I'm going to give or talk is just because focused on the just wheat. Next slide, please. And I always like to use this quote. I have drunk from wells I did not dig. I have been warmed by fires I did not build. Go ahead, please. The reason I use this is that science is a team research and really global. So what I want to say is that before I start my presentation, I like to acknowledge all my students and postdocs and research scientists, and as well as the master's students and PhD students, so that anything I'm going to give you or talk about genome editing or any other research is that because of my team. Actually, that I like to mention those, like I had 12 PhD students supervised, and they are all in different universities, 16 master's students, and almost like 20 scientists and postdocs. They are all in different universities around the world, across the world. I really enjoyed working with them. And I just wanted to say that anything I say is just I'm representing them and is coming from my team. Next slide, please. So in this, and you can continue as well, that I gave some numbers, World Health Organization reports in 2017. I was going to ask, like, if you were in-person talks, that I was going to ask, what are those numbers? Do you have any idea that if I ask you the question? So can you please go ahead? Next slide. Yeah, how many people are really undernourished, and then, or if there's any hungerness? And the report says, go ahead and please, 11% of the world's population is undernourished. So it's about 820 million people. So 22% of the children under 5 years old is stunted, like they don't grow really well. There are a number of reasons. And that there's a severe food insecurity as well. 9% of the population that they have under this category. And if you were to look at the last one, is 1 in 4 people globally is really having difficulties for food insecure. So the insecurity and then is totally different than that the availability of the food. I'm not going to go into detail, but these are the real numbers that we really need to think about, that how we can just focus on these numbers to reduce that so that people can get the food or available for them, or they will secure it. Next slide, please. So there are, if you were to look at it, there are top 5 factors of the world hunger. I can talk about each one of them: poverty, climate change, war, conflict, and food shortage, and then poor nutrition. But as a scientist, and I always share this information with all of my students and postdocs and scientists and my team actually, and 2 are the most important thing that we can really focus on and we can do something. Although maybe it's going to be indirect contribution. Number 1 is that the climate change. I will talk about a little bit about water deficit and drought stress as well. And number 2 is the poor nutrition, which means that increasing the nutritional effects and the nutritional value of the wheat or any other plant species, so that for instance, kids or children is going to have enough nutrition. Next slide, please. Okay, if you were to look at it, this is the number that we need to be really, really careful, that water in, on, and above the earth, if you were to look at it, that the small amount of a circle of the water is the water that we can use. So that's the water that we are using in the whole wide world. Next slide, please. But we are consuming this water because, like, for food, for instance, this is a cup of coffee. We all love to drink a cup of coffee in the morning. I wish I did have a cup of coffee right now. But do you know that how much gallon of water that we spend for this, just one cup of coffee? Next, please, which is about like 34 gallons of water that we are spending. Not only for food, when you look at the next slide, please, that for industrial aspect as well, that we are spending a lot of water too. For instance, for a single white t-shirt, we are creating 700 gallons of water that we are spending to have this t-shirt. So we are losing or using that water for food, industry, and agriculture, which I'm going to talk about what we can do about the agriculture, and then how we can apply to one of the breeding tools, which is genome editing, to reduce the water deficit or drought stress. Next slide, please. However, working in wheat is really big challenge. It's not easy. If you were to look at this slide, you will easily see that looking at the Arabidopsis genome, which is 140 megabases, 10% of the repetitive elements. Brachypodium, we as a team with the international team, we sequenced all of them, 355 megabases, and which is like small. Human genome, I will talk about 3,000 megabases. Think about it. And then this is the 6 times bigger than human genome. This is the wheat. And not only like a big genome and polyploidy, but also like a repetitive sequences. If you were to look at it, like more than 90% of the genome is repetitive sequences, which we are trying to understand that how they regulate the genic effects. So this is a big challenge right now. That's what we are focusing on, that how we can deal with it, and think about that how we can apply the CRISPR system or any other breeding tools to wheat genome. Next slide, please. And there's, I think that I haven't heard from the other speakers, but I just wanted to say that there are 3 main genome editing tools, not only CRISPR, but the zinc finger nuclease is number 1, ZFN, and TALEN are another one of the genome editing tools. However, CRISPR, I'm not going to go into each one of the details, but it has a higher efficiency. CRISPR has a higher efficiency, and it has a low cost compared to ZFN, zinc finger nucleases, or TALEN. And then it's really rapid, and multiplexing is possible. I will give you some information and our recent research about the multiplexing. And there is low off-target effects as well compared to other editing tools, which we are using for plant system already. Next slide, please. And I will not really go into detail because the CRISPR system is very well described in detail. Previous speakers really did a great job. Thanks to them. I mean, but I just wanted to let you know that this is what we are doing for wheat. Basically, that we are really applied this system and the flow chart for wheat plants. And first thing that we are trying to understand what the target gene is we are focusing on. I was giving you some examples about the drought and water deficit. So we just, in our first experiment, we just took 2 drought responsive genes. So we wanted to edit those genes and try to set up the protocol for wheat system. As I said before, it is a huge genome and polyploidy, and it's not easy to work with. So that's why we wanted to set up that protocol for wheat only. Next slide, please. And then we started with the vector construction. And I will just give you 2 examples, and everybody talk about the Cas9 system. What we use is the Cas9, CPF1, Cas12 as well. So that I will give you some example that how Cas12 and Cas9 works and what are the efficiency in wheat system, because that you can apply to that CRISPR to human genome, insect genome, that we are focusing on insect genome as well right now. So that we wanted to understand that which one is really most powerful, effective, and efficient. So this is the Cas9 system, but the right picture is clearly showing that the CPF1 as well. Next slide, please. I think I don't need to talk a lot about the guide RNA design and vector construction because previous speakers already talked, but in wheat, we just look at that the PAM sequences as well, as we all know. And then Cas9, SP-Cas9, Dr. Özcan was talking about the NGG is that the essential PAM sequences, and CPF1 that we use the TTTN, and CPF1 as well, that the two nucleases that we use the same guide RNA. And then we can just design a CRISPR design or Cas designer. We can design the guide RNA using these two systems. Now, I just wanted to mention that I didn't want to mention here because I had some slides, but I didn't know that you guys are going to completely put it out. And we have some machine learning system and deep learning system. We did the algorithm. Hopefully it'll be out soon, in a month or so, that we will publish. We have we applied the machine learning system to design the guide RNA for wheat, just wheat and other cereals as well. So, and then we are just selecting the target gene and synthesize the guide RNA. Go ahead, please. Next slide, please. So this is one of the experiments that we did, and then we used the different promoters, and then we analyzed that the 3 target gene, and you can just see that the ERF1, one of them, DREP2 is the another one, and we designed the guide RNA using different promoters. Next slide, please. Okay, that one of them is the Oryza sativa, the U6 promoter. We use this different promoters so that how it's going to affect the wheat. Next, please. And there is a maize promoter as well, UBI. And the next, please. And triticum U3 as well that we used as a promoter, and then in that construct. And then looking at each one of the sequences, we designed the guide RNA. And based on the promoter, you can see that the PCR screening and guide RNA oligos and then PCR primers were designed based on this. Go ahead, next slide, please. And another one, 6, yeah, as well. Go ahead, yeah, let's just, yeah. So what we did is that everything is going, we put it in the one construct vector, CRISPR Cas9, and this was the first experiment to set up the protocol. And then we look at the ERF genes, which is drought responsive genes, and DREP as well, so that we wanted to identify or edit those 2 genes, see that if it's going to work or not. Next slide, please. Yeah, and then U6, and then guide RNA was put it in the construct, and then scaffold was there, 35S promoter were there at the end, Cas9 and LS as well. So we designed the primers. When we isolated, we looked at the PCR results, and everything was working perfect. Next slide, please. So the important part is that how we can deliver or introduce into wheat genome. As I said before, it's a huge genome, and then not every gene delivery methods is going to work perfectly fine for that. But we do have a next slide, please. In wheat, there are 3 main things, tools that we can introduce any genes into plants or wheat, actually, I would just say. And one is the bioelastic particle bombardment, which is easy, but there are some pros. Each one of them has a pros and cons. So we need to be really, really careful that what we are really doing. And bioelastic system is really good. I mean, we did that. We used that for introducing that vectors to wheat plants. But sometimes there are a lot of cell damages, and then we couldn't understand that the expression level of the genes that we introduced. And there's a PAG-mediated chemical delivery. I will give each one of the real example that we did in a bit. So this is good, high efficiency, but the generation of the plants is really hard after you bombard or you transfer the genes. And agro is the really good one, which we just easily just target the one region of the genome, and then we can put it that gene or vector that we constructed, and then it'll go there. However, in wheat, the efficiency is 1 or 2%. I mean, we tried 10 or 12 different elite varieties, and then you can look at the efficiency is really, really low because of the huge genome and number of other things that in wheat, that the regeneration problems and other stuff as well, that we had the problem as well. So go ahead, please. And this is the PAG method that we just published in 2018, how we can just deliver the protoplast isolation and gene delivery for constructed Cas9 system. And basically that we are just collecting the plants, isolating and cutting and cleaning. And then at the end that we are just calculating the seed cell counting, and then under microscope that we looked at it, and then 5-6 hours that incubation that we will look at that, what the enzymatic digestion as well. Well, as you know that the cellulase and pectinase is critical here when you do the protoplast isolation or when you use the protoplast method. Next slide, please. And however, that in wheat, after that we constructed everything for the Cas9, and then we looked at the GFP expression, and based on the GFP expression, you can see that in 24, 48, 72 hours that we just got like 70-80% of cell counting, and so we were really lucky. And then we said, okay, we will go with the protoplast bombardment for Cas9, introducing the genes using Cas9. So that was our first experiment. Go ahead, please, next slide. And then we also did the particle bombardment, and then you can see that we recently published in this on the right side, phenotype after bombardment. You can see that if you can see that the gene, what we did is that we used the PDS gene. And PDS gene is that the bleaching type of gene. You will see that if there is some kind of albino type of plants. So when you added that PDS gene, which we did, and then we saw that what are the efficiency of the CRISPR-Cas system using particle bombardment. And then we used the 3 target genes, and then you can look at the albino plants phenotype using Cas9. You can see that under the picture, and then CPF1, AS, and LB-CPF1. So that we wanted to understand that which Cas9 system is really best, or I would say the better for wheat plant. Next slide, please. So this is just showing the particle bombardment. Yeah, go ahead, next slide, please. So then agro is the one that I believe that we should go with the wheat. And based on the results that we have currently, that we are just the callus generation that we are getting, and then after transformation, we are going in selection process and plant regeneration. And then we really identify the 1 transgenic plants, which means that we introduce the gene vector to the plants, wheat plant, and then we can just utilize and characterize the plants. And this is, again, that when I say plant, it's just a wheat. Go ahead, please. Next slide. Yeah. And what we did is that this is the vector construction, again, that the CRISPR system, that what we used that in the system, Cas9, CPF1, ASCPF1, and LBCPF1 as well. This is coming from the different bacterium. So all we did is that we used the different promoters, and then we would put the Cas9 right after the NLS, and then guide RNA, and then NLS, and then nose term as well. So that this was the vector construction, 3 different vector construction. And we just simply introduced that guide RNA, again, the PDS gene, so that we wanted to edit just the 1 gene and to compare all 3 of the nucleases as well. Yep. Next slide, please. And after that, we need to validate that. I mean, based on the wheat genome, there's a number of validation that we were talking about. And one of the validation that we were using is the T7 endonuclease assay, which is like basically if there is a mismatch in the genome right after you introduce the gene or vector, that it's gonna cut the place that where the endonuclease is going to cut the repair part, and then you will see that the differences between that and regular or wild type, or that if there is a cut, like this is the target 1, for instance, you can see that there was a cut in different genes or fragments that you can easily see that there was a cut, so that you can easily say, okay, T7 endonucleases is showing that the differences too. Next slide, please. And then we also didn't want to stop just looking at the T7 assay. We also did the Sanger sequencing, although it's just the time, it takes some time to understand that what the sequencing is going to come, and Sanger sequencing. It's not expensive currently. I was listening one of the talks. It's getting much cheaper and cheaper now. And what we did is that we sequenced, and based on the sequences, and wild type, we easily identified that this genes is just mutated in different, using different nucleases, CRISPR nucleases. Next slide, please. And based on all of the data that we analyzed, using T7 enzyme and then CRISPR secondary sequencing, what we identified is the LB-CPF1 was the higher efficiency compared to the other ones. Like using target 1, you can look at it, SPA-Cas9 was the 18 plus minus and 2%, and then 19 in using CPF1, use ASCPF1. But look at the yellow part is mainly that higher efficiency. So we decided to use LB-CPF1 and for editing some genes in wheat, and not only that this target genes, but also that we are focusing on some microRNA editing as well. There are some microRNAs. We were just really successful for some prime editing of some microRNAs too. Hopefully that we will be able to get the results and publish in that everybody is going to see that the results. So this is what I like to say for that. The next slide, please. And I just wanted to mention that multiplex genome editing in wheat as well. We had a couple of genes that we wanted to just edit, especially for micronutrient genes in cadmium in durum wheat, which is like we are making pasta out of it. Is the cadmium level is always high, and there's a codex number that if you wanted to just import or export that, you have to look at the cadmium level. And if it is really high, nobody is going to buy, and market is going to be really low for that durum wheat varieties. But it is really hard to deal with that cadmium gene. So what we did is that we identified the cadmium gene, since we sequenced the whole wheat genome, and then not only Chinese spring, but also like a 10 wheat genome is being sequenced, the pen genome wheat is out right now. And then we just look at some cadmium genes. And we were focusing on that if we decrease the cadmium, it's going to be good for exporting that type of elite varieties. But also, there's another important issue is that when we decrease the cadmium, the zinc level is going to increase. So zinc genes is going to be increased, which zinc is really important for wheat as well. So what we did is that we designed guide RNAs for number of cadmium genes, and we put them like 2-3 different guide RNAs and put it in this one vector, which we said like a shuttle vectors that we are using for each guide RNA. At the end, we are combining in one plasmid. Finally, that final destination vector is that containing all the guide RNA and Cas9 too. That's the red one. Next slide, please. So we did that. I mean, we just look at that 3 different regions that we guide RNA. We put it in the one vector. Eventually, final vector is ready, and now we redo this whole thing again, using that guide RNA and everything, because previously that there was a little bit differences. So that's why we just wanted to use, redo whole experiment again. And then now everything is ready for the single vector construction is ready that we are going to introduce into wheat, hopefully in a month, so that we will understand that if we can reduce the cadmium level in durum wheat, so the exportation or export is going to be easy for farmers, growers, or NGOs or other clienteles. Next slide, please. Well, I just said that the challenges in wheat genome editing. There are 3 main challenges, I would say, is that the main focus is number 1 is that the huge genome and polyploidy level, which is there are 3 genomes in one wheat whole A, B, and D genome. So when you design one target, sometimes it's impossible to get rid of the off target, but also like you can target the different genome and same gene as well, because same gene might have 3 copies in each one of the genome, A, B, and D. So that's one of the difficulties in polyploidy level. And then huge genome and then repetitive sequences is another challenge. And then third is that the efficiency of the transformation, like agro is the one is really, really low, 1 or 2%. I've never seen that like more than 3-4% of efficiency that you can easily transform. This is difficult in wheat. So that's why maybe one of the things that I would like to mention right now here is that there is no GMO wheat in the world. So people were really confused about like saying, hey, there's a GMO wheat, but there's none. So those are the challenges that we are trying to deal with in wheat when we are editing. So one of the things is that I will mention, I was going to mention that I had slides that we did the prime editing in microRNA and in base editing, so that it was a little bit different than the whole target gene because of the small part and everything. So it was really hard in wheat. So dealing with wheat genome is really, really hard. But I want to mention also, like, there's a whole reference genome sequencing is available, and you can easily target the gene, identify the gene in wheat now. Next slide, please. And I just wanted to mention that the machine learning for genome annotation as well. This is critical. What we started is that we were manually annotating, or we were using totally different tools to annotate the genes. What the gene is, what are the targets is. And what we decided recently is, because we are really dealing with the non-coding RNAs and so mainly long non-coding RNAs and microRNAs, what we decided that we can write the algorithm and to identify and separate the non-coding RNAs. That I'll give you one example that we published in this, which is long non-coding RNA. And then we started like looking at the machine learning system and wrote, and it is available right now, is that some algorithm. First we discussed and then analyze whole algorithm, and then we wanted to add another algorithm to see that which one is going to be best for annotating long non-coding RNAs or microRNAs, and using machine learning. And then if you were to look at it, what we are doing is that for machine learning, as everybody said, described, and we had a training set, and some of them are unsupervised. We did that as well, or supervised too. And then we did the machine learning algorithm, put it in there, and we the machine just look at the features of the long non-coding RNA and separate it. You can see that the blue one is just looking at the separation of the long non-coding RNA. But we also put it in new data to the machine so that we can just, or algorithm, so that we can identify the totally different one as well. Next, please. Yeah. Okay, but you can put the next as well, so that I will discuss everything together. Yeah, and one of the critical part is that the false positive and false negative too. So this is one of the rock curve, and what we are really evaluating is just if we can just get like a much more true data instead of like a false positive or negative, false negative data. That was one of our issues or challenges, I would say. But this was the issue that in wheat genome, especially that a separation of long non-coding RNA. However, that some of the tools I will mention in a bit. Go ahead, next slide, please. Next slide, please. Okay, that helped us a lot in terms of like a long non-coding RNA annotations. For instance, that we used it like a random forest, Adobe Boost, and then native Bayes and native Bayes and QDA and things like that, and whatever that available currently, so that we can easily just tell them apart about the efficiency, training accuracy, and so that was important. And based on this, what we have right now is that the LNC machine, we put the algorithm and data to GitHub as well, that everybody can go back and look at it as well. So it's also like we published in Functional Integrative Genomics recently, and if anybody is interested in, they can look at it too. This is just, again, that the mainly wheat but we also compared some other data sets to, like Arabidopsis too. Next slide, please. So that we, based on the wheat data set, we also use the different ploid level, like tetraploid wheat and hexaploid wheat as well. These are the like references that we just use, and then it's in the published part. But you can easily see that the long non-coding RNA differences in hexaploid and the tetraploid wheat based on or using machine learning too. Next slide, please. I just want to mention like a little bit about the mirror machine too. This was another one for microRNA annotation. I will not go into detail, but the microRNAs are small. MicroRNAs are the small part of the region of the genome in the mRNA, and you can look at that. If there's a mismatch, and we can just, dicer is going to cut, and we can identify, and looking at the first structure, secondary structure, and also like first sequences and secondary structure and tertiary structure as well, so that we will identify it's a real microRNA and what gene that is targeting, which is really, really important in terms of regulation of the genes. Maybe there's not direct relationship for the gene expression, but the expression of the microRNA is giving us the regulation of the genes and leads to the regulation of the genes. Next slide, please. So here's the, yeah, we are just looking at the mirror machine. This is, again, that it's in the publication recently. Hopefully that we will have a video for that from the Jove, and they already contacted us, but we didn't have a chance to just do the video. That we will go into detail and explain each one of the detail how we used. Now that we are applying some of the algorithms to mirror machine so that we can easily automatically annotate that the microRNAs, and so that it's going to be like out there hopefully soon. But we're working on that. But the blue ones is mainly showing that the wheat, and then some orange ones are showing that the Arabidopsis data set that is available if anybody is interested in. It's been published in Jove as well, that if you like to go and look at the data and then results as well, additional results. Go ahead, next slide, please. And this was our early data analysis that we used the different wheat varieties. For instance, Kızıltan, one of the Turkish lines, TR39477 is that another wild lines. TTD, again, the wild wheat as well. And these are the like most drought tolerant and drought resistance lines that we used. And we look at the networks analysis as well, that this was clearly showing that each one of the lines based on the drought or water deficit showing different color. And then you can see that there's some purple color here, and then here is that mainly showing that some lines are really resistant. On some of them is that they're not resistant or just less resistant. Next slide, please. And I like to end up, finish and conclude with one of the really African proverb. If you want to go fast. If you want to go fast, go alone. If you want to go far, go together. And I just added the last sentences from me. If you want to go fast, oops, and far, go together with using integrated tools and technologies. We've been talking about the CRISPR new technology and like a machine learning, deep learning, and new algorithms, and so that we need to use whatever available, all kinds of tools and technologies to fight with the hunger, to fight with a number of other issues like diseases and other stuff, the other issues or problems in the world, using all kinds of technologies, whatever available currently. With that, I'd like to thank all of the funding opportunities, again, that the funding sources from USDA, NSF, NIH, like some Montana local funding as well, and then Montana Bioag funding too. And I'd like to acknowledge that. And also I'd like to acknowledge all of my team members, previously and current team members as well. With that, I'd like to thank you again, and I'd be happy to address any question that you might have. Thank you.
Thank you, Professor Budak, for the very nice speech and the presentation. That's really a very interesting topic as well. When I check, okay, the time management is also perfect for everybody.
Good.
I can say this one, that exactly at the time. So if for any questions that anybody want to ask you, I think they can reach you.
Sure.
So, okay. Thank you very much again for your time, for your, for your valuable information you just have given. We are thankful to you again.
I thank you.
Okay.
All right.
Thanks, Özcan. See you. Okay, now, as everybody knows, at the beginning we had a connection problem with Dr. İlker Gelişen, and now we will continue with Dr. İlker Gelişen. The problem was solved, and then with his talk we can continue. The title is Interaction of RNAs, or antibodies, I guess, with coronavirus. And let me introduce him again. Dr. İlker Gelişen graduated from Hacettepe University, Faculty of Medicine in 1977. After specializing in anesthesia and reanimation in Germany between 1978 and 1984, he worked in various positions, including general manager in the pharmaceutical industry in Turkey for 30 years. Since 2013, he has been teaching basic medical sciences, anesthesia, and artificial intelligence as a lecturer at nearest university. His special interests are digital transformation in healthcare and drug discovery and development. He is the Turkey representative of the International Federation of Associations of Pharmaceutical Physicians and Pharmaceutical Medicine. I would like to welcome Dr. İlker Gelişen to the stage. Welcome, hocam.
So, thank you very much. As you realized, I did my best to be the first after the opening presentation, and then I also tried to be the last before the closing ceremony. What I wanted to do was actually to talk about something totally different than all other presentations. What I could not do from my room in the Faculty of Pharmacy in the Near East University, now I am doing from the studios of the Near East University. I mean, background has changed, but I am still the same one. What I'm going to talk about, or what I planned to talk about today, was actually something not totally new, but something becoming more and more important every other day. I started my presentation in the morning by talking about my university, Hacettepe University, in the center of Ankara, I can say. There is a road from Sıhhiye going to Hamamönü for the ones who know Ankara.
I'm sorry, hocam. Could you please make it full screen, your presentation?
Just a second. It is now full screen.
Yes, thank you very much again.
Is it better now?
Yes, perfect.
Thank you very much. Then in my old days, which goes back to the other century, when we started learning microbiology and our lecturers started talking about coronaviruses, I put a note, not important, not related to the healthcare of the human beings, and it was true for decades. Common human coronaviruses have circulated in the human population without any significant mortality. But especially end of 2019, which is the name of the COVID-19 actually, severe acute respiratory syndrome with the coronavirus 2 is associated, and it became a part of our lives. And we all have heard a lot about it. For the coronavirus, there is, I mean, when we come back to or concentrate the situation or the thing I'm going to tell you is that for the coronavirus to get inside the body, the human body, there must be an interaction between a protein that is on the surface of the virus, which we call it the spike protein, which we call as also S protein. This protein has to bond to a receptor protein on the human cell receptor we know is called as ACE2. I mean, full name is angiotensin converting enzyme 2. That is on the surface of our bodies. I mean, when we talk about where do we have those receptors, I mean, in the respiratory system, in brain, in heart, in kidneys, in liver, in the gastrointestinal system, etc., etc., all around. It's only after this interaction that S protein, that the virus has, the virus can enter human cell and forcibly direct it to spread the viral RNA and causing a cascade of cell infection. That is the famous coronavirus. We all have seen it a lot of times. I'm sure we can draw it when we close our, even when we close our eyes. Those are actually the S spikes, which give the coronavirus the view of a crown, and that is the reason of calling it the coronavirus. Here, what I'm doing is a short video which explains the core of the I mean, the problem I had has not solved, but I will tell it. That is what we try to see in the video. I will try it once more, but I think the problem is continuing. Moving from the faculty of pharmacy to the studios did not help us. The thing is that the coronavirus has S spikes, and we in our body have angiotensin 2 receptors. Coronavirus, after entering our body, must have an interaction with the ACE2 receptors, bind them. When it binds, there are a couple of actions. I'm not going to go in details. It was clearly shown in the video, and after the presentation, maybe you can look at it. It is a very nice video showing how the interaction is. Then the virus goes close to the body, can enter the body, and after the virus enters the body, then actually everything continues. Those are S proteins you see.
Hocam, there is a problem, I guess, with video, so the sound is not coming and your voice is not here when you use the video, so maybe you can explain it without video.
That is very, I mean, what I said can be easily seen in those two videos. That is the coronavirus entering our body and then reaching the areas where we have ACE2 receptors. When they bind them, that is the most important stage. After binding ACE receptors, then a couple of other actions happen, and the virus can enter our cells. When it is in our cells, it starts replication, etc. Then that is the disease. The spike protein, therefore, is crucial for the coronavirus to infect healthy human cells. The spike protein is the main target of multiple COVID-19 vaccines and antibody drugs. That means if you can block it, if you can do something against that S spike, that protein, then you can prevent coronavirus entering the body. It will come and go then without causing any kind of an infection or any kind of a problem in the human body. Simply by blocking this interaction between spike protein and ACE2 receptor is a very effective way of neutralizing the entry of coronavirus to the human host cell. Then we may think of strategies. What can we do? I mean, what are we doing? What are we trying to do now? The vaccination programs, vaccines we are trying to prepare, the therapeutics, the medications we can do against it. They are mainly, most of them are targeted to the S spike. Meanwhile, the researchers, while dealing with it, showed that there are nanobodies which appear to trigger a key structural change when they bind to the virus. This change is likely to be irreversible. The virus is therefore no longer able to bind the host cells and infect them. That is one of the most important targets we do now. What are those nanobodies? In late 1980s, from Belgian, Raymond Hemmers and Cecil Kesterman had discovered a class of antibodies that were devoid of light chains and had a single antigen recognizing domain. These antibodies were later found in different camelid species, including llamas and alpacas. I mean, if we talk about the story, those two researchers have biology students give a homework to get antibodies from the species they have. The students were a little bit, I mean, they did not intend to do it with the human blood because of HIV, one of the most important problems during those days. They did not do it with mice, and they found some actually important parts of a camel in the refrigerator, and they started looking at it. And they found those small molecules. They were actually small molecules, apart from the antibodies we have, and they were effective. The researcher then injected a piece of SARS-CoV-2 spike protein along with the inactivated virus into one alpaca and one llama to elicit the production of antibodies against the virus. Among the millions of antibodies generated by the animals, the team eventually picked 4 nanobodies, 1 of them from llama and 3 from alpacas. That shows best ability to bind and neutralize the virus in the laboratory dishes. Those nanobodies, look, you see on the right side of the slide, they are small, they bind irreversible, and therefore the virus can no longer bind to the host cells and become the reason of the infection in the human bodies. Nanobodies may hold an advantage that in their small size, they are small-sized molecules. Therefore, they can penetrate deeper in the tissues. Nanobodies are also more stable and are easier to produce, potentially make them less costly. Those are actually the advantages we can say. Then, when you go through, when you search, you can see a lot of people, a lot of companies, a lot of organizations have interest. Many scientists believe could inspire treatments that offer advantages over traditional antibodies. Researchers from the University of Bonn, the Karolinska Institute, the Scripps Research Institute, they designed nanobodies based on drugs for COVID-19. Another company, Dioscure Therapeutics, has spun off from the University of Bonn with plans to bring its lead to COVID-19 candidates, Dios 202 and Dios 23, and they are planning to do it bringing it to the clinical trials in the course of 2021, this year. University of Texas, Austin, Belgium, I mentioned, they are all working in those directions. Twist Bioscience reported recently a report, which is late May 2021, that 2 of its nanobodies protected harm cells from weight loss, which is an indicator of COVID-19 disease severity, at a similar level to that of plasma from COVID-19 survivors. The last thing I'm going to talk about is actually the origin, what I got that idea, what I heard that idea, what I started thinking about is the University of California, San Francisco, UCSF. They posted encouraging results. You see the names of that publication. I started counting and ended up with 34. 34 different people, and OCRG Structural Biology Consortium is one of them. And the last two, Peter Walter, Ashish Maglik, are actually the heads of that presentation. They called their nanobodies, and they want to do it as aerolized antibodies, put in sprays and use them in sprays. And they designed it and are willing to block the S spikes with those nanobodies when actually we think that when we have an exposure to COVID-19, the coronavirus, or we think we have just contacted, or we want to travel, then having some kind of a spray and using that spray with the help of those aeronaps designed by UCSF, it will be possible to block S spikes, and we will contact the coronavirus, but the coronavirus will be blocked by the nanobodies, here aeronaps, and will not be able to enter the host cell, our cells, and cause the infection. When we talk about them, I mentioned in another slide, actually, they are stable. It is easy to produce them. Therefore, they are cheap. We can self-administer them. We can have the spray with us. And when we think we have it, or when we may have it, we can use it. We can, there are 3 entrance ways for the coronavirus to human body. Those are mouth, nose, and eyes. And the most common ways are the nose and the mouth, and we can spray it there. Therefore, we can directly deliver the aeronaps to the area where we think or where we suspect of coronavirus. And we can dose it as we wish it. As a summary, I want to point out 3 facts, as almost all presenters did today. The world is changing faster than ever. From the Harvard University, there was a presentation in November 2020. The presenter was talking about the half-life of medicine. That means the amount of time we need to get half of the information, we cannot use it anymore. It was actually 5 years in 1990s. That means there was a nice presentation of the Dean of the Harvard University Faculty of Medicine. In the opening presentation of the school, he was telling it is getting faster, which makes the half-life of medicine 5 years. That means in 5 years of time, the amount of information I know at the moment, half of it will not be valid. The problem I have is that which half? And in the presentation in November 2020, the presenter was talking about the half-life of medicine again, and he was talking of 73 days. That is a very short time. Every day we are producing 10/18 bytes of new information. Reading them, knowing them, learning them is not easy anymore. Therefore, we need to come together, do things all together, like the aeronauts, 34 different people, 34 different background come together, do it. And in terms of our work in the universities now, I mean, I'm the only one in all those presentations, not from the Faculty of Engineering. But we need to be more and more, we need to understand each other better and better, work more and more together, and be helpful to other human beings, like the aeronauts against the coronavirus. Thank you very much for listening to me. I am very happy you like to ask questions. If not, all I wanted to deliver today.
Okay, thank you very much, Dr. İlker Gelişen, for the very nice presentation. So just I want to say that as your final comments, we should do more frequently such kind of activities as the lifetime of medicine decreases. So we should we should also decrease the time interval between such kind of activities, I guess. So thank you very much again.
Thank you for giving me that opportunity, because I need you.
Thanks, Hocam. Thanks. Okay, so again, I would like to thank to all participants for their and also all listeners for their valuable time. And I would like to invite Professor Özsöz to do the closing speech. So, Professor Özsöz. Okay, maybe
it's okay
there's
now.
a problem.
No, it's
okay. Yes, Hocam. Welcome, Hocam.
Okay, thank you very much, Süleyman Hocam. I really appreciate all your efforts and also the contributors, you know, and also technical staff for the contribution to this meeting. And once more, I say that, you know, what we have seen, you know, here in the talks, you know, the power of knowledge is very, very important, you know. So the only way that, you know, we can go further and further was education, and the knowledge, you know. So the peace will become, you know, if the people are educated and if they got knowledge, enough knowledge. That's very important. And one of the speakers that Hikmet Hocam that mentioned about how can we eliminate this problem of food, you know, in the world is thus, you know, the new technology, and just give this technology to the poor people who they need it, you know. So I want to once again thanks to the nearest university that he gave us this opportunity to meet with these listeners and with the speakers. So to meet in the future meeting, thank you very much for everybody.
Okay, thanks.
Thanks.
Catching a bus, never nothing when it's real, yeah.